Dear colleagues, welcome to this training for the instrument of pre-accession. And you are very welcome to this webinar, which will start with a very interesting topic on a new product evaluation. First, I want to give you a few housekeeping rules. Yeah, c'est bon? We have because we use WebEx, you need to unmute yourself. You need to mute and unmute yourself when not speaking, and when you want to take the floor, you need to unmute. Some of you may be double muted because you also have a mute function on your microphone, for example, so don't forget to double unmute yourself, but mostly don't forget to mute yourself when you're not speaking, because otherwise we have a lot of background noise and it disturbs everybody. But you're very welcome during this webinar to take the floor and to ask questions. Preferably raise your hands, because then we can see who wants to speak and give the floor in turns. Alternatively, you can use a chat function and put your question in the chat function. And with Caroline, who is going to give you the webinar today, we will take the question on an ongoing basis or during the normal breaks of this session. Please don't worry, you will receive the slides after the training. Because we have a number of questions in the slide, we don't give you the slide before, but you will have the slide afterwards. So no worries, you don't need to copy everything. This will come to you. Okay. Anything else? So I'm not going to go around the table for everybody, because we will get probably more than 100 or 200 participants. We also have a few assessors from the EU national competent authorities who are also welcome to this training, and members from the European Commission. But I would like to make sure that we have at least one participant from Albania. Could you please say if you're there from Albania? Do we have Jetmeri or Navina? Okay, I will get back to you later on. Do we have someone from Bosnia and Herzegovina?
Yes, Jasmina Bradic is here from Federal Ministry of Agriculture.
Thank you, Jasmina. Thank you for confirming. And I hope you have many of your colleagues with you. Thank you. Do we have a Kosovo? Do we have Ariane or other from Kosovo or Valdet? Okay, I have nobody for the time being. Montenegro, please.
Hello, from Montenegro, Željka Bešlić here. Me and my colleagues are here.
Thank you very much for confirming. Thank you and welcome. North Macedonia?
Yes. Hello, everybody. This is Todor Karapancha from North Macedonia.
Thank you very much for confirming and welcome. Do we have someone from Serbia?
Yes, hello. So greetings from Serbia and Medicines Medical Devices Agency. I'm sure we have colleagues from Ministry of Health participating as well. Thank you.
Thank you, Pavel, and welcome. Do we have people from Turkey, please? Okay, maybe not yet. Can I go back to Albania? Do we have someone from Albania? And do we have someone from Kosovo? Okay, we will confirm later on who is participating from these two countries. In the meantime, I suggest that we are moving on. And Caroline, I'm giving you the floor to start the presentation. And first, let me thank also Clément and Sandra, who you don't see for the timing, or you see in the general view of the room, and they are supporting this webinar. And thank you very much for doing that. And thank you for the audiovisual support as well. Thank you. And as you know, Ricardo is not here, but he was the one preparing most of this webinar. Thank you. Okay, Caroline, you have the floor.
Yes, thank you, Anais. So it's with great pleasure today that I'm going to give you a presentation about the evaluation of an initial marketing authorization application. But before we start, I would like to ask you a few questions. So Clément, if you can put the Slido. So it's very easy to use, so don't worry. So basically, you can Google slido.com. You can enter the code, so it's hashtag 1932, and I will be asking a question, and you can respond directly in the Slido. So Clément, can you put the question? Don't worry, you can still see the code.
Just for the participant, you can either use a smartphone to get to Slido and answer on your smartphone, or you can use another computer to get to Slido. It's quite easy once you're there. So, and we keep on the left-hand side of the screen the QR code, and also if you go directly to the by computer, you have the hashtag 1932 to join. Thank you.
Okay, so in your views, what are the most important aspects to consider when evaluating a dossier? So first response, eating an apple a day keeps the doctor away. I prefer oranges. Practicing sports, playing the piano, having someone you can rely on, being kind to others. Or last response, I'm not sure I understand. I thought I was going to attend the training on the evaluation of medicines. So as you can see, there is no wrong answer, so don't be shy. I need just to break the ice this morning. So we keep on so that more responses can come in. A bit more, 13.
These questions are also intended to help you use Slido first.
Yes, indeed. Yes, okay. Clément, shall we try to see the responses, and hopefully more questions will come, more response will come? Yes, that's the response I was hoping that you will say. So, and it was just to break the ice, so don't worry, it's nothing to do with the evaluation of medicines. Yeah, so maybe we can move on to the real questions. So in your views, so Clément, can you put the slide? So in your views, what are the most important aspects to consider when evaluating a dossier? So I would like you to think about the main aspects. So it's free text, so you can put whatever you want. And we will have this question at the very beginning and at the end of the presentation. So again, don't be shy. So all the responses will come as a cloud. So we will see most of the responses as a cloud. So it will be quite interesting to see your answers. And you can still use the same code. So it's the same question. Don't be shy. You can put what you think is most relevant. Can be quality, non-clinical data. Yeah, good. Safety and efficacy, completeness, quality. Yes, accuracy, quality. More responses. Yeah, I think people now enter responses. That's really good. Benefit risk, that's good. Consistency, low. Yeah. Completeness. Okay. Okay, I think let's stop. And then we will have the same question at the end after the presentation. So as I can see, the most answered questions are quality, but also quality, safety and efficacy, which is and balance risk-benefit, deep analysis as well, accuracy. So that's already very good. Okay, so maybe let's move to the presentation. So Clément, you can maybe put my slide. So I believe I can also push the slides if you give me the host, Clément. So next slide, or can you put me as a host so that I can, yeah.
You are the host now.
Yes, Now I am those. Yeah. Very good. So I will first start with the main consideration with regards to the evaluation of a dossier, and then I will be using crizanlizumab, which is also called ADAC-VO, as a case example. And before I start, I just would like to mention that, of course, all aspects with regards to the evaluation of crizanlizumab are only a choice of selected topics and for the purpose of a training. So for the full comprehensive review, you can see the European Public Assessment Reports. And I also would like to confirm that there is no confidential information in this presentation. So where to start? So I believe at the very beginning it looks like a giant puzzle where you don't really see which first pieces you need to connect. There are a lot of things to see, and it's not so easy to understand where is the starting point. So usually the starting point is actually the legal basis, which is also defined in the EU legislation. And depending on the legal basis used, so for example, if it's a full marketing authorization application or a generic, this will define the data to be provided. Usually, quality non-clinical and clinical data follow scientific guidelines that are published on our website. We have also specific guidelines on the risk management plan. And the output of the evaluation is the product information, the risk management plan, and of course, the European Public Assessment Report, which details the evaluation of the medicine. So first, it's all about context and also perspective. So it's really important to understand what is the target indication, and in the target indication, what are the available treatments available. And of course, depending on the product's type, for example, if it's a tablet or an injection, it will mainly change the posology to be given, the efficacy outcome, and also the safety profile. So the evaluation of a dossier, it's also a two-dimensional approach. So first, it's a comparison versus the products already authorized in the same indication. And for this, usually the efficacy aspects are looked at in terms of efficacy endpoints, efficacy results, which are usually provided in section 5.1 of the product information. So when applicable, so in the context of a conditional marketing authorization, so this is a marketing authorization which is granted when we define specific post-authorization safety and efficacy studies called specific obligation that have to be imposed as part of the marketing authorization and will be submitted post-marketing to switch the conditional marketing authorization as a full marketing authorization. And in this context, we have what we call the major therapeutic advantage, where the medicine is assessed in comparison to products authorized in the same indication. In the context of orphan medicine, we have what we call the clinical significant benefit. And again, this would be an assessment versus the authorized medicine products which are already authorized. It's also very important to look at the differences in product information in labeling if the product is already authorized by other agencies. And with regards to the comparison versus products class, it's very different on the quality aspects if you have a chemicals versus a biological. First, you have very major differences in the manufacturing. But also on the class effect with regards to the safety aspects. So, for example, a monoclonal antibody is very different to a gene therapy, and it's also very different to blood products. So we know that there are specific safety aspects for certain type of products. So, for example, monoclonal antibodies may have injection site reactions depending on because of the route of administration. For gene therapy, we know that for retroviral vectors, there is a risk of insertional oncogenesis. For blood products, for example, there is a risk of immunogenicity and neutralizing antibodies. So it's also very important to know the biodistribution of the medicine, the route of administration, and that will lead to defining specific warnings, contraindications in the product information, and when applicable and possible, to find additional risk minimization measures, and when applicable, to have specific post-authorization safety studies to further assess the risk identified during the evaluation of the medicines. Just to say, it's also important to look at the new active substance status, and the orphan similarity. So both concepts are very different, but have to be also looked at in conjunction. There are 3 pillars, and that's something you have already identified as part of the question to assess a medicine. So quality, non-clinical, clinical, and safety and efficacy. And that leads to the product information and the risk management plan. So with regards to quality, there are 2 parts, the active substance and the finished product. So with regards to the active substance, it's important to define very well the manufacturing, the characterization of the active substance and the process controls, the specification, the stability. For the finished product, it's also a description of the product. So to be sure that we can define well the product, that will lead to the pharmaceutical development, its manufacture of the product and the process controls, again, the product specification, the stability of the product, advantageous agents, and when applicable, the genetically modified organism, which have also to be assessed. So with regards to the non-clinical data, so you have the main aspects, so the pharmacology, so the proof of concept, defining the mechanism of action, having the secondary pharmacodynamics, the safety pharmacology, the pharmacodynamic interactions, the pharmacokinetics, toxicity, and the environmental risk assessments. I would like to point out for the non-clinical, usually this really serves as a very good basis to define the safety profile of the medicine. And then with regards to the clinical data, so we have usually the PK studies, which usually define the dose. The phase 1 study defines the tolerance and the safety of the medicine. In some cases, the phase 2 clinical trial can already determine the clinical efficacy. Most of the cases, it's used to look for the optimal dose of the medicine to be given. The phase 3 is usually the pivotal clinical study, which will support the marketing authorization. So all in all, the efficacy in the context of authorized medicines with the quality and the safety profile will determine the patient's treatment. So it's really important to characterize the efficacy in the context of the authorized medicines with the quality and safety profile in order to understand the place of the medicines in all the treatments available for the patients. So in order to help companies to develop their products, we as EMA have different tools available. The Innovation Task Force is one for innovative medicines where companies can have a briefing meeting with us in order to ask specific questions. We have the small, medium-sized enterprise office. When applicable for advanced therapy medicinal product, we have 2 procedures called the ATMP classification, so to define whether a product is an ATMP or not. We have also the certification of quality and non-clinical for ATMP, so it's some kind of evaluation of quality and non-clinical data. We have, as I said before, specific guidelines, so it's really important to look at the available guidelines, which will determine the data to be provided at the time of marketing authorization. So when applicable, we have also the orphan designation, so specifically for orphan medicines. We have the qualification of novel methodologies. And again, when we have a pediatric indication, then the companies need to submit a pediatric investigation plan that we call PIP to the pediatric committee, which is called the PDCO. Last but not least, we have the scientific advice where companies can ask specific questions on their development in order to have some input on the development of their medicines before the marketing authorization. And then we have the priority medicines, which is a scheme for companies which will have an enhanced regulatory support, and that is given usually for promising products.
Caroline, maybe we can make a little break here because it's a lot of information. And I see a hand raised by Slobodanka. Slobodanka, would you like to take the floor, please? If others have questions, it may be the right time to ask your question. Slobodanka, can you unmute yourself to speak? You have raised your hand. Okay, it doesn't seem to work. We cannot hear you. We cannot hear you. So we will move on. Caroline, maybe you can just put an overview of all this pre-approval procedure, because it's quite a lot of possibilities. maybe summing up a little bit that some of these procedures are necessary, others are optional?
Yes, indeed, and yes. So as you pointed out, some are optional. So it also depends on the type of the product. So for example, for orphan medicines, so if your product has been designated as an orphan medicine, then of course you would have to apply for the orphan designation. In the case your product is an ATMP, you have the possibility, so that's again not mandatory, but you can apply for the ATMP classification and the certification of ATMP quality and non-clinical. Again, the Innovation Task Force is not mandatory. It's usually here to help companies when they have innovative products. They have specific questions. It's usually for academia who are not really familiar with the process of developing a medicines, and they would like to get first views on the development, and that's really used as a first step for this applicant to start with the development of their product. Again, the SME office, it's also very specific to small and medium-sized enterprise. And again, the pediatric the scientific advice and the priority medicines, it's also not mandatory. It's usually recommended for companies to apply for scientific advice because, of course, they will gain a lot of support in the development of their medicine. But with regards to the pediatric investigation plan, this is mandatory. So all companies need to come to the PDCO to get a pediatric investigation plan. And that is no matter if there is no pediatric indication or if there is not even the possibility to have a pediatric investigation based on the nature of the medicine, they will get a waiver, but they have to apply to the PDCO to get this pediatric investigation plan agreed. And that's based on the pediatric legislation in order to support the evaluation and the approval of medicines also in the pediatric indication. Then I have not mentioned before, but when the product is then assessed, we have the possibility to have accelerated assessment, and again, that's usually for products which are promising, and then the assessment timeline is reduced from 210 days to 150 days. And this is, of course, to help putting innovative and promising products on the market as quickly as possible. The conditional marketing authorization I have alluded before, it's specific for, so it has 3 criteria. So it's usually when the medicine is indicated for life-threatening disease, and then there is an unmet medical need, and the product represents a major therapeutic advantage. And in this context, when the benefit risk is positive, then the CHMP, so our main committee, which is called the CHMP, can grant a marketing authorization. And we will ask specific post-authorization safety and efficacy studies that we call specific obligation to be submitted, so that after 5 years, the conditional marketing authorization will be switched to a full marketing authorization. Maybe I will now go to, unless there are any specific questions, but otherwise I will move on to the case example. So it's crizanlizumab, also as a trade name Adakveo. And this product is indicated for the sickle cell disease. And as you may know, sickle cell disease is an heritable disease caused by a single missense mutation in the beta globin gene. So usually it's mainly prevalent in sub-Saharan Africa, the Mediterranean area, the Middle East and India. And the EU prevalence is quite low, so it's below 2 per 10,000 individuals. So that is the reason why it has been classified as an orphan medicine, as it fulfills the criteria of orphan designation. The management of sickle cell disease usually includes the treatment of pain episodes and its clinical manifestations that can be life-threatening. In general, patients have severe acute chest syndrome and stroke. Patients get treated with transfusions, and in some cases, splenectomy can be necessary. Hydroxyurea or hydroxycarbamide is a standard of care, and it can decrease the frequency and the severity of vaso-occlusive crisis. It usually reduces the transfusion needs, but many patients do not tolerate it so well, and they still experience veno-occlusive crisis. The only curative option is hematopoietic stem cell transplant, and it follows a myeloablative conditioning regimen, but it's only limited to children with a matched sibling donor, so it's not applicable to all patients. In some cases, and most recently, match unrelated and haploidentical donors is an alternative method used, but is not the current standard of care. So all in all, and considering the available treatment, we can consider that there is a medical need in this indication. So the initially proposed indication for Adakveo was for the prevention of vaso-occlusive crisis in sickle cell disease and in patients aged 16 years and above. Finally, the CHMP approved the indication for the prevention of recurrent vaso-occlusive crisis in sickle cell disease patients aged 16 years and older, but it can be given as an add-on therapy to hydroxyurea or hydroxycarbamide, or as a monotherapy in patients for whom hydroxyurea or hydroxycarbamide is not appropriate or inadequate. And as I said before, Adakveo was designated as an orphan medicinal product in the condition of treatment of sickle cell disease. So with regards to the pediatric indication, so the company, as I mentioned before, submitted a pediatric investigation plan to the pediatric committee. They have a waiver for the children below 6 months of age and a deferral for patients or pediatric patients above 6 months of age. So it's expected that they will complete the clinical studies and the pediatric investigation plan by September 2023. And it's quite interesting in this case because the PDCO asked the company to perform an open label study, but also an extrapolation modeling and a simulation study that will support the granting of a pediatric indication. So moving on to quality. So that's the first pillar of the evaluation of a medicine. And of course, there are a lot of things to say, so I've tried to summarize what are the critical parameters when evaluating quality aspects. So at first, it's very important to have a robust and consistent manufacturing process, especially for biological medicines. It's important to have process validation data, which will ensure that the manufacturing process reliably generates active substance and the finished product according to predetermined specification and quality attributes. Again, the characterization of the product is really crucial, especially for biologicals. And what happens often is companies usually changes their active substance or finished product, so they have two different versions of their product based on enhancement of their quality of the medicine, and that leads to perform some comparability exercise between different processes. Potency is also very important because it will influence the efficacy aspect. It's of course very important to remove any impurities as part of the manufacturing and assess any adventitious agents. The nature and the content of container can have some interactions with the finished product or even the active substance when applicable. So it's always very important to assess if there are any interactions with the container. Last but not least, the good manufacturing practice. So of course we can conduct inspection as needed. So we have what we call the routine inspections, where we inspect manufacturing sites, especially for new sites. But we can also have a triggered GMP inspection as part of the evaluation of a medicine, and this is usually defined when assessors assess the dossier and think that there is an inspection which is needed in order to confirm certain aspects of the evaluation. So usually the quality will determine the recommendation for use of the medicine. In this case, the stability was 18 months. There is some special precaution for storage. So, for example, the medicine has to be stored in a refrigerator. It cannot be freeze, and the vial needs to be kept open in the outer carton in order to protect. The vial needs to be kept in the outer carton in order to protect from light. Usually, it's also very important to understand how the product is going to be prepared and administered, especially to give recommendations for healthcare professionals. So in terms of calculation of the dose per body weight, how to administer, some specific precaution for use. And in this case, there was a recommendation to address specific issues post-marketing, and the company has to reevaluate the current specification for the active substance and the finished product based on the commercial batch manufactured. So we forget.
Maybe before we move to the next session, can we ask if you have any question or a point you would like to clarify? Because it's, again, a lot of information in one go. It's a complex product, so of course the evaluation of quality is also complex. But if you feel that you would like to ask a question, please take the floor, unmute yourself and take the floor, or raise your hand or put the question in the chat. We are monitoring the chat so we can see if you have a question. Sometimes it's easier than taking the floor. So I hear no voice. I see no hands raised. Nothing in the chat for the time being. So please continue, Caroline. Thank you very much.
Thank you, Agnès. So with regards to the non-clinical part, so it all starts with the mechanism of action and the pharmacology. So crizanlizumab is a selective humanized immunoglobulin G2 kappa monoclonal antibody that binds to the human P-selectin with high affinity, and it's blocking the interaction with its ligand, including P-selectin glycoprotein ligand 1. And the elevated levels of P-selectin was found in patients with sickle cell disease. Hence, P-selectin is a cell adhesion molecule that plays an important role in the development of vaso-occlusion and veno-occlusive crisis. Crizanlizumab was shown to be species specific, and it only binds to P-selectins from human and related non-human primate and monkey species. As part of the manufacturing, the human SLG1 antibody, which is crizanlizumab, was initially developed by a company called Reprexis, but upon acquisition by Novartis in 2016, several drug substance manufacturing changes were made, including a cell line switch, a manufacturing process changes, and a formulation change, resulting in a new version of the monoclonal antibody called SG101. So to start, of course, it's important to understand what are the animal models to be used and what is the choice of animal use, which would be the best to determine the non-clinical program. So primary pharmacodynamics were only studied ex vivo for SLG1 and SLG2, so the two versions of the antibodies, due to occurrence of immunological reactions and species differences in target recognition of P-selectin. So no preclinical in vivo studies in a sickle cell disease model or in a vaso-occlusive model with the humanized antibodies were done with the two versions of the antibodies. And because we had two versions of the antibodies, the company did the bridging data between the two versions, and the target recognition for the two versions of the antibody was based on an in silico primary sequence analysis of selectin isoforms and on an in vitro surface plasmon resonance binding assay, and used to bridge between the two versions of the antibodies. So in the non-clinical program, it was found that crizanlizumab interfered with the ex vivo platelet counting assays in human EDTA blood. Therefore, there was a recommendation added to monitor these aspects and the platelet count in section 4.4 of the SmPC, which is a specific warning. So this is a busy slide, but it shows all the non-clinical safety pharmacology and toxicology studies that have to be performed for a medicine. So it usually starts after the mechanism of action and the proof of concept, so pharmacodynamic studies, with the safety pharmacology. which is usually used in order to see if there are any effects on cardiovascular, central nervous system or respiratory function. And in this case, this was evaluated in 4 weeks and 26 weeks repeat dose studies in cynomolgus monkeys. And in this case, no effects were found in cardiovascular, CNS or respiratory function. The pharmacodynamic drug reactions was not conducted, but it was assumed that the simultaneous use of crizanlizumab and anticoagulation treatment might lead to an increased risk of bleeding. And the potential risk of additive effect was taken into consideration and was assessed as part of the clinical safety part of the dossier. So with regards to toxicology, so that has been performed according to ICH6 R1, which is a preclinical safety evaluation of biotechnology-derived pharmaceuticals. And there was a single toxicity study conducted and the 26-week repeat dose toxicity studies conducted. An enhanced pre- and postnatal development toxicity study, tissue cross-reactivity study conducted, and cynomolgus monkey was chosen as anti-P-selectin antibodies were demonstrated not to bind to murine P-selectin but only to monkey. The NOAEL was determined to be 25 milligram per kg per dose. So no genotoxicity and carcinogenicity were conducted, but that's according to our guideline, ICH S6 R1, again, which doesn't require for these studies to be conducted in these cases. So with regards to reproduction toxicity, it was found that crizanlizumab may increase fetal loss or stillbirths in animals treated, and that will be addressed in the clinical part. So the local tolerance was assessed in the repeat dose toxicity and enhanced pre- and postnatal development toxicity studies. So with regards to the environmental risk assessment, no environmental risk assessment study was conducted, but that's acceptable because it's in line with our specific guideline, which states that medicinal products containing proteins due to the nature that are unlikely to result in a significant risk to the environment. So it's very important to note that when applicable, some specific studies could be used for different purposes. So that was the case for these 4-week and 26-week repeat dose toxicity studies, which were used in order to define the toxicology, but also the safety pharmacology. And again, the local tolerance. And that's to, of course, avoid as much as possible the use of animals in non-clinical development. So the non-clinical pharmacokinetics and also the clinical pharmacokinetics. So in terms of non-clinical data, so the pharmacokinetics of the two versions of the antibodies was compared in a bridging study after single dose using 2 doses of 5 milligram or 10 milligram per kilogram and administered to cynomolgus monkeys. The clinical data was assessed in 251 subjects, 87 which were healthy and 164 patients with sickle cell disease. And they derived from 4 clinical studies analyzed to characterize the crizanlizumab pharmacokinetics based on the population PK approach. So pharmacokinetic studies usually aims at giving guidance on absorption, distribution, metabolism, and elimination. And that's usually the guidance we provide in section 5.2 of the product information. Again, it's important also to define the differences in special population, and that can be, for example, patients with an hepatic or renal impairment, differences in terms of body weight or age, that can be, for example, for the elderly. It will also determine if there are any interactions. In this case, the effect of a concomitant medication, including hydroxyurea or hydroxycarbamide, corticosteroids, anti-inflammatory agents, was examined in a population PK approach, and it was concluded that concomitant medications, including hydroxyurea or hydroxycarbamide, had no effect on crizanlizumab pharmacokinetic in patients with sickle cell disease. So all the information about the pharmacokinetics will lead to the posology, which is stated in section 4.2 of the SmPC. And usually we mention whether a treatment should be initiated by physicians experienced in the management of sickle cell disease. So that's what we call restricted prescription. So not all healthcare professionals can give Adakveo to their patients. So the recommended dose of crizanlizumab is 5 mg per kilogram, administered over a period of 30 minutes by intravenous infusion at week 0, week 2, and every 4 weeks thereafter. And based on the pharmacokinetic studies and the interaction studies, it was mentioned that crizanlizumab can be given alone or with hydroxyurea or hydroxycarbamide. So moving on to the clinical part. So the main study was called the A2201. That was the registration study. And that was the study used in order to support the marketing authorization of crizanlizumab. Before the company submitted the dossiers, they applied for protocol assistance. So that's what we call scientific advice, but for orphan medicine. And they asked specifically whether the proposed study used as a post-marketing safety and efficacy study will be adequate in order to generate sufficient evidence to fulfill or to address the specific obligation in the context of a conditional marketing authorization. So they ask specific questions about the design, the evaluation of 2 doses, the study population, the primary and secondary efficacy endpoints, the definition of vaso-occlusive crisis, aspects on statistical consideration, sample size estimation, for example, and the adequacy of the evidence to be generated to support the conversion of a potential condition marketing authorization to a full. They also ask the adequacy of the envisaged clinical evidence base to support the significant benefit. So that is in the context of the maintenance of the orphan designation, because at the end of the marketing authorization, the COMP, so the Committee for Orphan Medicinal Products, will determine whether the orphan indication can be maintained and the product should be still an orphan medicine. And that is conducted after the evaluation of the marketing authorization application. So the main study, called A2201, was a multi-center randomized double-blind parallel group placebo-controlled study evaluating the efficacy of 2 doses of crizanlizumab with or without hydroxyurea in sickle cell disease patients aged 16 to 63 years of age. Patients included any sickle cell disease genotypes, and they had to have an history of 2 to 10 vaso-occlusive crises in the previous year, and they were randomized in a 1:1 ratio to 5 mg per kilogram crizanlizumab, 2.5 mg or placebo. So the randomization was stratified by hydroxyurea and the number of vaso-occlusive crises in the year preceding the study start. So basically, the patients served as their own control. The study consisted of a 30-day screening period, a 52-week treatment period, and a follow-up period of a maximum total duration of 58 weeks. The primary endpoint was the annual rate of sickle cell-related pain crisis. The key secondary efficacy endpoint was the annual rate of days hospitalized. So
Maybe
the company...
Caroline, we
yes.
can make a little break here again because of the amount of information. We still don't have questions or in the chat or raised hands, but I think it would be good to try to maybe extract what is specific here. We are in a disease which is not so frequent in Europe. It's mostly around the Mediterranean Sea, as you said. Can you extract what is striking here, the type of control they've used? I would like also to emphasize the fact that the primary endpoint is a clinically relevant endpoint that is meaningful for the patient, because maybe after that we can skip the, also to keep the time for discussion, skip a little bit some of the details and go for the, but again, in terms of teaching how to assess, what was the main point you consider relevant in this main pivotal trial? Thank you.
Okay, Thank you, Agnes. So indeed, it was shown that the primary endpoint was clinically relevant because it's also very important to understand what is the patient's main value of having a medicine, and the sickle cell related pain crisis, as we can understand, is something which is very meaningful for the patients, and also the key secondary efficacy endpoint, which was the rate of days hospitalized, meaning that the crises were severe enough for the patient to go to the hospital. So I won't go maybe into the details of all the statistical analysis, but just to say that there were several statistical analyses provided, especially in the intention to treat population. So that's usually the statistical analysis that we look at, and the supplementary analysis which are provided during the evaluation used investigator-based vaso-occlusive crisis data. These were the ones that our committee rely on, and they excluded one outlier patient because it can also happen that one patient is an outlier and may not be needed to be taken into consideration in the statistical analysis. Yes, in terms of main aspects, I think it's really important to first understand whether there are any differences in the baseline characteristic of the patients. We cannot compare apple versus oranges, so all the patients included in the crizanlizumab arm should be more or less the same patients as in the placebo group. And that's, as you can understand, is really to make sure that we can compare the two groups adequately, and there are no differences at baseline which will influence the outcome of the treatment. So in terms of efficacy results, so the primary endpoint was the annual rate of veno-occlusive crisis. For ADAC-VO, it was 1.63. For placebo, it was 2.98. So the p-value was 0.010, so it met its primary endpoint. And again, as I mentioned before, the primary analysis, which was supplemented by several analyses, including a negative binomial regression or investigator-assessed veno-occlusive crisis, with a conservative method to handle missing data was used, and the response rate was 0.74. So the primary endpoint was met. So it's also important to look at the consistency in the efficacy results. So, for example, all other endpoints, in this case, there was a clinically significant reduction in the annual rate of veno-occlusive crisis across important subgroups. So these, for example, whether hydroxyurea or hydroxycarbamide was used in the previous months, also across genotypes. And that's really reassuring because it shows that the efficacy is consistent across the subgroups. So there was uncertainties and limitations of the favourable effect. The first one being that there was analytical differences between the product used in the pivotal clinical phase 2 study. So as I mentioned before, that was the version of the antibody developed by Reprexis and SG01, which was the one manufactured with the commercial manufacturing process and by Novartis. But however, the impact of the differences on in the product quality on PK and efficacy and safety was rather unlikely. And the preliminary data that we received from study A2002, which used the commercial product, was comparable to study A2201, which used the previous version. So we thought that the two versions of the antibodies were comparable. But as I mentioned before, since it's a conditional marketing authorization, additional results will be submitted as specific obligation to the marketing authorization, and the study A2002 will be submitted as a post-marketing commitment. As part of the evaluation, it's important to note that there were critical GCP findings, and it was considered that the evaluation of treatment effect based primarily on the Center Review Committee adjudicated data was not considered sufficiently robust. However, the data collected at the study sites were credible, and the analyses which were provided during the evaluation, so the ones using the investigator-based veno-occlusive crisis rates, were considered acceptable for the benefit-risk assessment. So this is the reason why, despite having seen the critical GCP findings, our committee considered that this efficacy analysis were considered acceptable for defining the benefit risk. And since it's a conditional marketing authorization, additional results of studies will be provided as specific obligations. So there were uncertainties again with regards to the statistical methodology used. So neither the pre-specified nor the supplementary analysis, so the ones provided as part of the evaluation of the medicine, were suitable to reliably conclude on the magnitude of the effect. However, the negative binomial regression model, as well as the jump to reference imputation, were considered the most appropriate. And the reanalysis provided of the effect of crizanlizumab in patients with and without hydroxyurea or hydroxycarbamide treatment. So these analyses requested to assess the therapeutic advantage of crizanlizumab or in addition of the authorized treatment were considered acceptable. So moving on to the unfavorable effects, and usually we always have drug-related serious adverse drug reactions, drug-related adverse events, and in this case, the main unfavorable effect was infusion-related reaction, which can be expected because it's a monoclonal antibody, and also an effect on the hemostasis system. So therefore, when specific side effects have been identified, they are usually reflected in the safety specification of the risk management plan, where we provide the safety concern in a summary table. So in this case, infusion-related reaction was identified as an important identified risk, and effects on hemostasis, immunogenicity-related clinical consequences, again, it's a monoclonal antibody, and infections were put as an important potential risks, and missing information was the long-term safety and also the use in pregnant and breastfeeding women. So with regards to the uncertainties and the limitation of unfavorable effects, there was an unspecific binding to over-selectins, mainly the E and L selectin, potentially causing unwanted effects. But this was addressed by for the company to define adverse events of special interest connecting to the mode of action of P-selectin. And again, none were identified, but again, we will receive additional data as part of the specific obligation imposed as post-marketing requirements. There was an impact of 100% P-selectin inhibition. In the pivotal study, P-selectin inhibition was around 70%. Only limited data available from ongoing single-arm study 82201, so with the new version of the antibody, reached 100% inhibition. So therefore, the CHMP requested a reanalysis of the samples from study A2201 conducted with a new assay, and that indicated that the observed differences in the percentage of P-selectin inhibition between the two versions of the antibodies can most likely be attributed to differences between the assay used initially in the A2201 study and the new assay used in the new study. The updated safety data from the registration study, the A2202 study, suggested overall that there was a comparable safety profile across studies and only minor differences were observed. And again, additional safety data will be provided as part of specific obligation to the marketing authorization. So with regards to the uncertainties and limitations, yes.
Try to summarize a little bit also the lessons from the what was found and how it is implemented in practice by the CHMP. Thank you.
Okay. Yes, thank you, Agnes. So yes, then again, the uncertainty. So we saw that there were more severe infections and a higher rate of pyrexia. So pyrexia was seen in the context of infections over rates of infections balance. So this didn't raise really major concerns, but again, additional safety data will be provided. And that's the same. Sorry. Yeah, that's the same for the higher rates of events related to a potential effect of crizanlizumab on the hematostatic system. And again, there was no relation of the timing of the events with either the time on the study drug or time after last infusion. So this didn't really raise a major concern, but the CHMP thought that this could be further monitored as part of additional safety data provided and as a specific obligation to the marketing authorization. So crizanlizumab is a monoclonal antibody which is produced in Chinese hamster ovary cells by recombinant DNA technology. So because it's a monoclonal antibody producing CHO cells, of course, patients can have an hypersensitivity to CHO cell products. So therefore, this has been added as a contraindication in the product information. This is also the reason why it's likely that patients may have infusion site reactions, and the specific recommendation and warning has been added in section 4.4 of the SmPC in order to recommend and monitor patients for the side effects. Again, additional safety data will be provided in further studies conducted. So as you may remember, in the non-clinical study, it was shown that crizanlizumab interfere with the ex vivo platelet count assays in human EDTA blood. And for that, we added a recommendation in section 4.4 of the SmPC in order to mitigate the potential for laboratory test interference. So in order to avoid this risk, it was recommended to run the test as soon as possible, so within 4 hours of the blood collection, or use a citrate tubes, and that's when needed, the platelet counts can be estimated via a peripheral blood smear. So that's the reason why I mentioned that it's really important to have the non-clinical data, because they will lead at the end of the day to specific recommendation on safety in the product information. So with regards to the reproduction toxicity, again, in the non-clinical study, it was shown that crizanlizumab may increase fetal loss or stillbirths in animals treated with crizanlizumab. Therefore, as a precautionary measure, the PRAC, so that's a pharmacovigilance committee responsible for the evaluation of safety of medicine, has recommended that it's preferable to avoid the use of Adagveo during pregnancy and in women of childbearing potential, not using contraception. But in order to help determine the effects in pregnant women, healthcare professionals should report any pregnancies in what we call a pregnancy outcome intensive monitoring program, or PRIM, in order to monitor pregnancies and report any side effects as quickly as possible.
Maybe I can intervene here, because pregnancy is a topic that will come again in various presentations. I think it's quite important we realize that as regulators, we may not have done our job of ensuring that we have the proper data for women who want to become pregnant, want to know in advance what are the benefits and the risk of a treatment. Don't forget that leaving a disease untreated has also its own consequences. So it's quite important that we collect the data at the right time. The non-clinical data are very helpful, but we also need clinical data. So preferably we try to frame and monitor when the trials are done. But of course, if a pregnancy occurs while a woman is on treatment, and as you know, many pregnancies are unplanned, not necessarily unwanted, but unplanned, and the women may not know they're pregnant in the first trimester. So we need to make sure that there is a monitoring and a follow-up of the pregnancy. Natural losses of pregnancy occur in many women, but of course, if a medicine can increase the risk of pregnancy loss, stillbirth, this needs to be known, and we need to understand what is happening. So that is why this monitoring is very important. It should not replace the development of medicines for pregnant women, but it's a complement that we need to have, and it's quite important that every healthcare professional is contributing to the data. Sorry for interrupting, but it's a very important topic. After pediatrics, I think we have to work better on this topic. Back to you, Caroline. Thank you.
Thank you, Agnès. And I fully agree with you that this is really something important to monitor. So moving on to the next topic, which was the indication. So the final indication was for the prevention of recurrent vaso-occlusive crisis in sickle cell disease patients aged 16 years and above. And it was concluded that it can be given as an add-on therapy to hydroxyurea or hydroxycarbamide, or as a monotherapy in patients for whom hydroxyurea or hydroxycarbamide is not appropriate or inadequate. So in terms of context, as I mentioned before, hydroxyurea or hydroxycarbamide is the current standard of care for the prevention of recurrent painful vaso-occlusive crisis, including acute chest syndromes in adults, adolescents, and children. And usually patients taking hydroxyurea represented the majority of patients in the phase 2 study, so around 60%. No other medicinal product was currently authorized at the time of approval. So the rationale to change the indication was that the evidence was requested that crizanlizumab provides a major therapeutic advantage over hydroxyurea in the initially sought indication, so the prevention of veno-occlusive crisis. And the indication was restricted to add-on therapy to hydroxyurea or hydroxycarbamide, or the monotherapy if hydroxyurea or hydroxycarbamide is not appropriate or inadequate. So therefore, the revised indication targets the population for whom there is an unmet medical need and where it's claimed. And that's also the reason why the maintenance of the orphan designation was there, and also the clinical significant benefit. So this is the criteria used by the comp in order to assess the significant benefits. So with this revised indication, the comp also concluded that crizanlizumab also had a significant clinical benefit over existing treatment. So in terms of conditional marketing authorization, so as I mentioned before, there are different types of marketing authorization. So conditional marketing authorization is usually used when the benefit risk is considered positive. It's likely that comprehensive data will be provided as part of post-marketing safety and efficacy studies. It's a life-threatening disease, and there is an unmet medical need. So in this case, we knew that the data to be generated in the phase 3 study, so the A2301 study, so a multicenter placebo-controlled parallel double-blind clinical study in sickle cell disease patients 12 years of age and older, which was already ongoing at the time of marketing authorization, where the study, which will provide comprehensive data and scientific advice or protocol assistance, was given for the study design of this specific study. 240 patients will be included and randomized with a 1:1:1 to treatment of crizanlizumab. And it's important to note that a higher dose of 7.5 milligram per kilogram will be assessed and will be investigated in order to see whether it could further reduce the frequency of veno-occlusive crisis. So it's an open label extension study of 5 years. And the feasibility, so that's an aspect which was carefully looked at, whether the feasibility of the studies could be maintained. But it was concluded that as the study will be performed in countries where crizanlizumab is not yet marketed, the study could be initiated and finalized. And it's expected that there will be of 240 patients in 2.5 years. So in terms of unmet medical needs, so the second criteria to have conditional marketing authorization. So that was back to my previous slide in the context and perspective. So we need more effective and well-tolerated treatment for sickle cell disease. The current treatment is preventive, symptomatic, and in many cases not always well-tolerated. And further, as also mentioned before, despite the current treatment, many patients with sickle cell disease still experience veno-occlusive crisis. The curative treatment, so hematopoietic stem cell transplant, is only an option for selected young patients which have a matched sibling donor and is not the standard of care. So crizanlizumab exhibit a new mode of action compared to existing treatment, and that's offering an alternative to hydroxyurea as an add-on treatment option. So therefore, it was concluded that the unmet medical need is agreed in the revised indication. So the third criteria, which is the benefits to public health of the immediate availability, outweighs the risk inherent in the fact that additional data are still required. So sickle cell disease patients have a high unmet medical needs and few therapeutic options. So the immediate availability of treatment alternatives has the potential to reduce the disease burden, the incidence of acute complication, and consequently to extend the life expectancy and the quality of life for patients with sickle cell disease. Crizanlizumab showed a very favorable safety profile. So far, only infusion-related reactions were identified, and this was appropriately characterized and addressed via routine risk minimization measures. In these cases, a recommendation was given in section 4.4 of the SmPC to recommend the monitoring of infusion site reactions. Therefore, making crizanlizumab available to patients while still collecting comprehensive efficacy and safety data in the ongoing control phase 3 studies, so the A2301, is not expected to represent a risk to public health. And in this context, we had two specific studies. The first one, which was the A2301 study, and the second one, which was the A2202 study, where we will receive the final results of these two studies in 2025. And usually when we get the results, and if everything, of course, is acceptable, this is the time where the CHMP decides if marketing authorization can be switched to a full marketing authorization. In terms of take-home messages, I hope that it was important to make clear that the evaluation of a medicine is part of a context with the main question in mind: what will it bring for patients? It's really always important to understand what will be the value of a specific medicines to the patient. And again, looking at efficacy outcomes, but also in terms of quality. For example, the Posology can really have an impact, for example, on patients. Having a tablet versus an injections makes a difference for patients, but also looking at the safety and the side effects. So it's a data-driven evaluation based on 3 pillars, so the quality, non-clinical, and safety and efficacy clinical data. And it's and that's something which is really important, I believe, to me. It's a multidisciplinary approach, and as I could call it, a puzzle approach, because it has to be collaborative, as a quality part will, in certain cases, influence the safety profile of the study. It can also influence the efficacy, for example, the potency assay. So it's really important that things are looked at as an interconnected approach, as a puzzle approach, because things will always lead. So it's always important to have a very good connection between the assessors so that they can see things holistically in the evaluation of a medicine. And hopefully the puzzle will be more clear, and with all the interconnection, a nice picture could be seen, and the product can be well defined. And I would like to thank you for your attention. I would also like to acknowledge the evaluation team, so the CHMP rapporteur, the PRAC rapporteur, the CHMP chair, and the PRAC chair. So I would like to thank you very much for your attention. I don't know if you have any specific questions. Otherwise, we could move on to Slido and see if your responses have changed with regards to the main aspects important to consider for the evaluation of a medicine.
Thank you very much, Caroline. We will indeed move to the Slido and ask you again the question. Now that you've heard all the complexity for a new product, when we've on purpose chosen a monoclonal antibody to show you the complexity of the biologicals, as opposed maybe to simpler files such as generics or well-established use, which we will discuss later. But I think it's quite important that we go through this. And so again, please connect to Slido and enter in the open field. What do you think now, based on this, are the most important aspects to consider when evaluating? There is no prize for that, sorry. There is also no punishment if you don't answer the same as your neighbor. We are really looking at more having an idea of what you've retained from this presentation, which was quite dense. And so please feel free to express anything, including about the puzzle. Thank you. We have 160 participants, so we should get a few more questions, a few more answers, sorry. So while you were answering, thank you very much, everybody, for participating. This is what we want. We want something more interactive and more fit for purpose. So you have to tell us what you also have you got out of this presentation. And thank you very much, Caroline, because I think we've seen new concept coming up in this cloud. And in particular, we see the patient benefit, we see the legislation aspect, we see the aspect of post-authorization studies, but clearly the benefit risk and the three pillars that you very well identify are there. And I think this is, if I may, I think I have one question, so I will get to the question in 2 minutes. But I just wanted to sum up what we are doing here. If you look at the evaluation, it's actually a life cycle management of the product. So we recommend early interaction, either informally through the Innovation Task Force, the ITF, or more formally through scientific advice, or in the case of orphan drugs, it's called protocol assistance, but it's the same concept. We also want to have, in the evaluation of the three pillars, a relation between each of them. They are not independent. The quality, as Caroline said, will influence the posology. it will influence the way the product is administered, but it will also identify maybe the risk of having a product which is developed, and when it's scaled up, it becomes a commercial batch. It's actually a different product for biological. So the need to bridge the data from the investigational product to the commercial batch is necessary, and the follow-up is necessary. You can already identify some of the expected effect, because we know that monoclonal antibody, for example, raise issues of fever, pyrexia. And so these are elements that we know and we will monitor. Going to the non-clinical, I think it's quite important first to understand the pharmacology, but also to try to learn as much as we can from non-clinical studies, in particular studies in animals, to especially where you have very few patients, because we know that we will not be able to have a full picture because the disease is rare, at least in Europe. In the case of sickle cell, it's very common in Africa, but it's certainly less frequent in Europe. So knowing that we will have fewer patients in the file, we need to maximize the information from the patient, but we also need to maximize the information we get before we go to the patient in order to maximize the efficacy and the safety information. And finally, because we get a sort of partial picture of the product, we continue monitoring the efficacy and safety through the post-authorization study. So it's really a life cycle management of this very complex product. So I have a question, and I will read it. It says, is the conditional marketing authorization the only option available for medicines for unmet medical need? What are the other access tools available for this type of medicines? And how do you select? I'm adapting the question. How do you select the tool? Is it the company or is it the MA who does that? Caroline, can you try answering this question? Thank you.
Yes, thank you, Agnès. So there is also another route, which is called the marketing authorization under exceptional circumstances. And usually this application or authorization is based when there is a limited number of patients, so it's an ultra-rare disease, or when you know that it will be impossible for the company to generate the required data. So, for example, we had one recently which is publicly available. It was a product used for anthrax contamination. So it's impossible or difficult to give anthrax to patients in order to evaluate the safety and efficacy of a medicine to treat anthrax infection. So this is typically the marketing authorization under exceptional circumstances, when you know that you will not be able to get the data at the time of marketing authorization because of feasibility reasons, and then it will be granted under exceptional circumstances, but you will still have post-authorization studies requested. It could also be based on data from registries or literature, and that's another route where you can also have this applicable. And I think, Anyan, what was your second question?
Who decides on the tool? Thank you.
Yes. So usually the company can apply directly for the conditional marketing authorization or marketing authorization under exceptional circumstances. But it could also be that following the review, the CHMP may have a major objection on the evaluation of the efficacy and safety data, and ask the company to justify whether conditional marketing authorization need to be further justified. So usually it's coming from companies at start, but it could also be that the CHMP may request the company to justify the conditional marketing authorization during the evaluation, and this is usually because of major issues identified. So it's depending, but when a company applies for conditional marketing authorization, it's because they have also a strategy in plan. They want to put the market as quickly as possible, the product as quickly as possible to the market, and they know that they have already planned post-authorization safety and efficacy studies.
Thank you, Caroline. Just also maybe to explain that you can go for a normal marketing authorization if your product is meeting unmet needs. There is no obligation to go for a special type, but because we provide some sort of incentive benefits or special treatment, like for the prime, when we monitor we give, I would say, enhanced scientific advice. We take care of this very precious product. It's more an opportunity for you to benefit from the help and support from EMA rather than an obligation. And the conditional is, as was shown in the slide, the possibility to grant relatively early access. We have positive benefit risk, but at the same time, we see that giving access is outweighing the risk of not having the full picture yet. And we will continue with additional study post-approval. So it's again a benefit rather than an obligation. And the tool is there to facilitate and speed up the process. Do we have other questions, raised hands? Do we have people who want to intervene? Questions, comments, clarifications? Don't be shy. We're here for you. Okay, Caroline, from your perspective, what is the biggest lesson you've learned through the evaluation of this product, from your perspective?
Yeah, from my perspective, I think it's really the multidisciplinary approach. So I have been lucky enough to work in different parts of the agency, and I have realized, and I think I am put this message across, that a lot of things, for example, that you see in quality can really impact on the safety. And again, the clinical trials will determine the risk management. The regulatory parts are also important in order to make sure that things are done appropriately. So I think it's my lesson learned actually would be that things need to be looked at together. So it's an evaluation should not be static and having a quality non-clinical and clinical assessment, but the teams should sit together and see, for example, what are the main aspects that they see in their evaluation, and see how it has impacted or it will impact other areas. And especially for biologicals, I think this is even more important, because usually for biologicals, the quality would very much impact the safety and efficacy. So for me, the most important thing is really the multidisciplinary approach and the puzzle approach. So things have to be interconnected. So it's not all the assessors should sit in their own desk and see things individually, but more to sit together and understand each other in order to really make the puzzle complete.
Okay, we had someone who was unmuted himself, Riccardo Serafini. Do you have a question? Okay, doesn't seem so. So I have a question. What are the most common major issues to reject a dossier? Again, based on your experience rather than the theory, Caroline.
Yeah, I think it's usually it's not so much on the quality part, because the company should have the manufacturing process in place, the process validation in place. But usually, so if they come thus far, it means that the quality is already efficiently established. So usually it's more on the clinical results and the efficacy outcomes. So primarily having an efficacy which is not as good enough to grant a marketing authorization. Also, the safety, of course, is important, but it's again a perspective approach and context, putting into context. So it has always to be also in line with authorized medicines. But I would say mainly it's about the conduct of the study and the efficacy results. So it's really important to have the clinical studies well conducted, and of course that's something that the company can never predict, but having good efficacy results.
Okay, thank you very much, Karin. I realize the time has gone. Thank you very much to all participants. We reconvene in half an hour for the next webinar, which is about evaluating a dossier of a generic or a biosimilar. So again, that will be a bit of a contrast with the new product, but it will allow to see the links and the difference between the, thank you for the program. Yes, if you can go down a little bit. So we reconvene in half an hour, so don't go too far. But you have time for a coffee if you want in the meantime. And we will have one of our assessor from the Czech Republic, Tomáš Radimerší, and he will present in half an hour. Thank you very much, Caroline. It was fantastic. And I'm sure there will be questions after the session. And again, I just reassure everybody, the slide will be sent after the session for each of the webinars. Thank you very much, and talk to you in half an hour. Thank you. Dear colleagues, welcome back. We have our new session on generics and biosimilars presented by a colleague from the Czech Republic, Tomáš Radimerší. I don't want to mispronounce his name. He has kindly accepted to present to you his experience of assessing such products. So just a reminder for those who were not in the first session that if you want to take the floor, please raise your hand or and unmute yourself. You may be double muted. We had some problem hearing people. We have also sorted out the small issue with the chat. You can now chat to everyone. So just write in the chat your questions. Don't be shy. We really want this to be interactive. There will be some Slido question as well. So for those who haven't connected, please go to the Slido, slido.com and enter the number which was on the screen earlier, which is 1932. So hashtag 1932. So Thomas, I'll leave you now to present, and thank you very much again for doing this.
Thank you, Agnes, for nice introduction. Good morning, everyone. My name is Tomáš Radimerší. I'm not sure that I'm able to control slides.
You're a co-host, so you should be able to.
Yes, I am. Thank you. So I'm working at the State Institute for Drug Control in Czech Republic. I'm a head of non-clinical and clinical assessment unit, and in the next 30 minutes, I try to go through the really extensive topic, and it's evaluation of dossier for abridged applications. I try to focus on generic medicinal products, hybrid medicinal products, and also biosimilars. I understood that you had a previous session where the legal basis were discussed. So I hope that you are familiar with standalone applications and abridged applications. I saw the first presentation today, and I hope that there will be a little bit more questions to my topic. So if you have questions, please feel free to write in the chat or ask questions. I will try to answer all these questions or uncertainties. So if we move to the next slide, which is introduction of generics, and I will start with some definition. The generic is the medicinal product with the same pharmaceutical form and the same active substance with established bioequivalence to the reference product or to the originator. Generics are authorized according to Article 10.1, which is the article in directive, and reference medicinal products to which the bioequivalent should be built are authorized according to full dossier, which is 8.3 standalone application, 10A well-established use application, 10B fixed dose combination application, or 10C informed consent applications. These all can act as the reference medicinal product in bioequivalent studies. And what is important, that the generics are not allowed to come to the market earlier than 10 years after the reference medicinal product. To prove the equivalence between the generic and the originator medicinal product, the bioequivalence study should be conducted. This is the most common and standard way how to prove the equivalence between two products. And this equivalence is evaluated with respect to the speed and rate of absorption of active substance. And this is described by the plasmatic curve. As you can see in the second part of the slide, the red one and the blue one lines are similar. And therefore, we can say that these products are equivalent to each other. This is the aim of the bioequivalence study. As you see further, there is not always the need to make in vivo studies, but in general, this is the most common way how to evaluate the generic and how to authorize generic medicinal product. How to work with the studies or how to evaluate the documentation. We have several guidelines which we are using for the assessment of the dossier. The first one is the guideline on the investigation of bioequivalence. This is the most important and most commonly used guideline. Let's say we use it for all marketing authorization applications, and you can find there all needed or important informations. But you need to keep in mind that it is only guideline, and this guideline cannot cover all possibilities and or variants for comparison of test and reference product. Therefore, in some cases which are not described in this guideline, the companies come to the authorities and ask questions. How should we evaluate or compare this medicine with the reference product? Or what should we do if we want to authorize this medicine? And these questions, if they cannot be found in the guideline, it is up to us regulators to somehow evaluate the approach presented by the company and find a way how to establish the equivalence between test and reference product. Second one is the guideline for the modified release dosage forms. This is also briefly mentioned in next slides, but the modified release dosage forms are not so common like immediate release dosage forms. And last bullet is referring to product-specific bioequivalence guidance, which are specific guidelines or guidances how to prepare the bioequivalence study for specific active substances. This is prepared by working party by EMA, and this is focused on a lot of active substances with specific requirements. For example, if we should evaluate the parent compound or metabolite, if we should use a fat or fasted bioequivalence study, or how to proceed for evaluation of the results. So this is very useful for the companies and also for us as the regulators when we assess a certain active substance. And let's go to some specific slides regarding to the bioequivalence study. Let's start with the design of the study. We can say that on the picture you can see a standard design of bioequivalence study, which is crossover design, single dose study, which means that the subjects are divided into two groups, and each group receive either active drug or placebo in the first period, and after a washout period, the subjects are switched, and those who received placebo now receive the active drug, and who received the active drug in the first period receive the placebo in the second period. And each subject is a control to himself. So this is a standard design, and it's very useful for evaluation of bioequivalence. However, in the guideline, you can find several alternative designs, and these designs has their specifications for certain conditions. For example, for substances with long half-life, the crossover design would be not feasible, and the study duration would be too long. Therefore, the substances with long half-life are evaluated during the parallel design. Or drugs with high variability are usually evaluated during the bioequivalence study with replicated design. Here I have one example. This is a bioequivalence study with clopidogrel, and I have it on several slides to show you how the certain parts of bioequivalence study are usually or standardly designed and performed. So here you see the description of the study, and you can see that this is the open label, randomized, 2 treatment, 2 period, 2 sequence, single dose crossover study with healthy subjects under fasting condition. What is important to say that all, if it's possible, all bioequivalence studies are performed with healthy volunteers, and the condition under fasting or fat conditions depends on the active substance. Usually you should look into the SmPC of the reference medicinal product and follow the recommendation if the product is usually used with or without food. If there is no recommendation, you should perform a bioequivalence study under fasting conditions, because it's a most sensitive way how to compare differences between test and reference products. Another important part, as I showed, the crossover design is the washout period. And this washout period is a time between first and second period of the administration test and reference product in one subject. And this period should be long enough to avoid pre-dose concentrations before the second period. According to the guideline, this time between two periods should be at least 5 half-lives of the or 5 elimination half-lives of the active substance. In case of clopidogrel, where the elimination half-life is 6 hours, the minimal washout period should last 30 hours. In this study, the applicant chosen 6 days. I can say this is long enough and the washout period is sufficient. Another bullet here is sampling times and collecting blood from the subjects. And this is important to prepare the sampling times in a way to sufficiently describe the Cmax of the active substance, because it's important to catch the rate and extent of the absorption as much as possible. So this sampling times differs from active substance to active substance, and again this should be sourced from SmPC of the reference product, where the time when the Cmax is reached should be mentioned in section 5. Here is a useful table where you can find in all bioequivalence studies, and there is a lot of information about test and reference product. You can see here the strength of the tablet, dosage form, batches used as a test product and reference product. This is useful for comparison with dissolution profiles, where the same batches like in the bioequivalence studies should be used. Bioequivalence guideline also have requirements for batch size. It should be 1/10 of the production scale, or at least 100,000 of units. This is fulfilled here. And also a measured content is mentioned in a guideline. And there is a recommendation that the difference of active substance between test and reference product should be not more than 5%. This is also fulfilled here. And last but not least, the importance is of a source of a reference medicinal product, which should be in the European Union. Here is France, so it's also fulfilled. And now we are moving to assessment of parameters and results of the performed bioequivalence study. We usually assess primary parameters like area under the curve, AUC, from 0 to time t, or from 0 to infinity, and Cmax, which is a maximal concentration. Sometimes you find also Tmax, time of the maximal concentration, but this is not a parameter which belongs to primary parameters. The evaluation of these results is considered with respect to 90% confidence interval of geometric mean ratio. It means that a portion of test and reference product should be fully within the margins of 0.8 to 1.25 or 80% to 125% for standard bioequivalence study. These limits can be also changed. For narrow therapeutic drugs, the limits are from 0.9 to 1.111. This applies for AUC and Cmax, but also these limits can be wider for drugs with high intra-subject variability. But in this case, these margins can be widened only for Cmax. And for AUC, the limits should be the standard. It means 0.8 to 1.25. In such case, the replicated design of the study is recommended. Here you can see results of clopidogrel bioequivalence study. And if you look at the ratio for 90% confidence interval, and then you see that all numbers for AUC and also for Cmax are within the limits from 80 to 125%, and therefore the bioequivalence can be concluded. In some cases, there is no need to perform in vivo bioequivalence study, and there are some approaches how to compare test and reference product without the need of in vivo studies. This is called biowaiver, and this biowaiver can be applicable for different strengths of the same medicinal product. This is also used for comparison of different pharmaceutical forms, and there is also BCS-based biowaiver, and I will talk about it a little bit later. Biowaiver for strengths. This is used in case of several strengths is under the assessment or will be authorized, and there is a need for at least one bioequivalent study, usually with the highest strength, and if the active substance behaves similarly, linearly, it means that there is with the higher strength, there is a higher amount of absorbed active substance, and some general requirements are fulfilled, like the same manufacturing process, same qualitative composition of medicinal products, composition of excipients quantitatively proportional, and these strains behave similarly in dissolution. Then we can conclude that, for example, only one bioequivalence study is enough to prove equivalence for 4 or 5 strains of one active substance. Another biowaiver is for pharmaceutical form. This is used for solutions for systemic use, and for example for oral solutions in case of the same active substance in the same concentration and no difference in active excipients, for example like sorbitol or mannitol, we can conclude that the we can conclude on equivalence without the need of clinical or bioequivalence study. This also applies for medicinal products like effervescent tablets, because they are tablets, but at the time of administration, they are solution. So they can also apply for this type of biowaiver. Similar requirements applies also for rectal solutions. And in case of parenteral solution, there are differences between IV administration or intramuscular or subcutaneous administration. These requirements are in detail described in a bioequivalence guideline, and I will not go further in this. Last biowaiver is BCS-based biowaiver, and this is a specific case where certain active substances that behave or have a high solubility and high permeability, or at least low permeability. These classes 1 and 3 can be specified, or these active substances does not need to perform bioequivalent study, and dissolution profiles might be sufficient to prove equivalence between test and the reference product. This is further described on this slide. This is applicable for systemic medicinal products, immediate release oral solid pharmaceutical forms, and as I said, BCS classes 1 and 3. But this is not applicable for drugs with narrow therapeutic index, and this is also not applicable for medicines with pharmaceutical form as sublingual or buccal tablets, or those with modified release. For modified release medicines, there is a separate guideline. However, the general requirements are the same as for immediate release. Therefore, also for modified release products, the classic bioequivalence guideline should be taken into account, and a lot of requirements is described in bioequivalence guideline. One difference that I will mention here is the need for multiple dose studies. Specific requirements which products need to have which multiple dose studies is described in this mentioned guideline on pharmacokinetic and clinical evaluation. However, this is not applicable for those medicines that they have not or they have only low accumulation of active substance. And this will be all for generic medicinal products acting systematically. And here I have a few slides about the hybrid applications, locally acting medicinal products, or the products where the bioequivalence study cannot be performed. For these medicines where we cannot or we are not able to demonstrate bioequivalence, we need to make a therapeutic equivalence study and prove the efficacy and safety between test and reference product. This is not a very common situation when we assess the marketing authorization applications. However, from time to time, some such products are also applied for the authorization. So in case of therapeutic equivalence, there is usually a 3-arm parallel study comparing placebo, test product, and reference product. And there is not much detailed guidelines how this therapeutic equivalence should be designed, because it's individual and it depends on the characterization of the active substance, on the pharmaceutical form of medicines, and also for the target indication, because these equivalents should represent the approved target indication of reference medicinal product. What is different in contrary to bioequivalence study? In therapeutic equivalence studies, the patients are the preferred subjects, and these studies are larger, therefore they can be multicentric studies. And as I mentioned, this usually comes as a 10-3 hybrid application.
Maybe, Thomas, we can make a break here, if you don't mind, and ask if there are questions or comments or clarifications, because then we change a little bit the topic. So really, please don't be shy. Ask and take the floor if you want, or put your question into the chat if you cannot connect. But really, we need you to tell us what needs to be clarified or complemented, please. We have one question, which is, what about generic application where the reference product is not on the market anymore? Is this acceptable?
Well, this is quite a good question. It depends if we are talking about a new marketing authorization application for generic, and there is no reference product in whole Europe. Then I'm afraid that it's not possible to apply for generic, because we do not have opportunity to perform bioequivalence study or dissolution profiles. We are not able to compare this generic product to anything. So I'm afraid that in this case, it's not possible to apply for a bridge application, and another legal basis should be considered. For example, well-established use application, because I guess there will be some literature or maybe I don't know, 8.3 new 8.3 application with reference to some literature data, but without reference product there cannot be a generic.
Okay, we have more questions for you. The first one was, what do you do when the batch size for an orphan drug is smaller than 100,000 units, and they use bio-batch of manufacturing size at that time, for example, 40,000 units, and after marketing authorization, they decide to scale up to 100,000 as a variation. Do you accept the change, or would you insist on another bioequivalence study with a larger batch size? Thank you.
Well, I'm not a quality assessor, so it would be a little bit difficult for me. But I think in a guideline it's written, then if the company don't manufacture 100,000 units at the time of marketing authorization, they should use the whole batch size, which will be in this case 40,000. And for the marketing authorization application, it should be sufficient. However, how to proceed after the marketing authorization, it's the question on the quality assessors, and I don't think that they will in this case look at the bioequivalence guideline and question the amount of the batch size, but they will I think in the standard way evaluate this change of batch size, and they will not refer to this bioequivalence requirement. So I think it should be fine if it will be fine from the quality point of view. I hope it doesn't make sense.
Thank you, Thomas. I have a question from a veterinarian about bioequivalence for veterinary medicines, about biowaivers. When a generic product may be included as a waiver from bioequivalence study requirement for immediate release formulation, is this classification appropriate only for auto-generic product, considering that one of the conditions is the identical manufacturing process? This is a reference to a specific point in the guideline on the conduct of bioequivalence studies for veterinary products. If this is may require a longer response, maybe that's something you can also provide after the session, either in the chat or in writing. So feel free to postpone the response if it's too specific, but you may know the answer.
Well, my background is veterinarian, but I'm not familiar with requirements for marketing authorization of veterinary medicines, and probably I can look into these questions afterwards, because I'm not sure if I catch it properly. So if it's in the chat, I will look at it afterwards, and we can, or I can provide a written response if I find the answer, if it's okay.
Yeah, of course it is okay. There was something again about biowaiver for a veterinary product, but that I think should be answered at the same time. There is a question about what kind of studies are needed to prove therapeutical equivalence for nasal products, for example, a steroid nasal spray.
If it will be the hybrid application, the first way how to compare nasal products is from the quality point of view, to compare the composition in case of nasal products, if it will be spray, to also compare the device, or how to say, to be sure that test and reference product delivers the same amount of active substance. I think that there is also some comparison of clouds of these nasal sprays, but if all, let's say, pharmaceutical requirements are fulfilled, I think there is no need to perform a therapeutic equivalence study. In case that there will be some differences between test and reference product with respect to quality, I think there should be therapeutic equivalence study which will be focused on the proof of efficacy. And I'm afraid that I'm not able to say some primary endpoints at this time, but these endpoints, or this design of this study should correspond with approved indication of a reference medicinal product. I hope it's sufficient.
Thank you. I have another question. That's good. Now people have become awake, and they are okay. If there are a excipient which affect, for an oral solution, which affect the gastrointestinal transit and absorption, could you give an example of the justification for not doing a new bioequivalence study?
This is quite difficult, because as far as I know, there is not a lot of literature references that would describe the behavior of active substance influenced by these excipients. However, if the company is able to provide the justification referring to some small study or some literature that, for example, the amount of mannitol less than 5 grams per dose does not influence the absorption of, let's say, omeprazole or some active substance, then it would be a sufficient explanation based on some data. But if the explanation will be only we think that this is a small difference and it will not influence the behavior of active substance, it's not sufficient. But if it's based on a small study or literature references, then we can accept the differences in the amount of excipients in almost in all pharmaceutical forms.
A general question about the reference: is there a BCS list for EMA that people can use? I know there is one for
no.
the FDA, but
sadly, no. As far as I know, there is no list of all BCS based classification. However, if you will not find your active substance anywhere in FDA or anywhere else, it's sufficient to find in literature the description of dissolution profiles. And it is mentioned in a guideline. I didn't put it into slides, but there is a margin that if the active substance is released in more than 85%, it's considered completely released. So there are some recommendations, what means a high solubility, high permeability, and if you find or perform a dissolution study or dissolution test, you can say this active substance in this reference product behave like BCS class 1, 2, 3, or 4. So if there is no list of the active substances, you can find it in a literature or perform a validated dissolution test.
Thank you, Thomas. Another question relating to identity of manufacturing process for veterinary medicines, again, as a reason for a waiver of bioequivalence study requirement for immediate release formulation. There is mention that the waiver can be requested if there is identical formulation, meaning active substance and excipient, physico-chemical properties, identical concentration, dissolution profile, crystalline form, pharmaceutical form, and particle size distribution with identical manufacturing process. Can you clarify what is meant by identical manufacturing process in that sentence?
Well, again, I'm not a quality assessor, but I guess it depends again on a type of active substance or pharmaceutical form. But for me, identical manufacturing process would mean that it's at least in an ideal way manufactured in the same facility, or it's I think or medicinal products have quite in detail described the manufacturing process. Each step is validated, is described, and for regulators it's easy if they have the documentation of a reference product to compare these manufacturing processes. So I think it depends on the evaluation of pharmaceutical assessor. I'm afraid that I cannot be more specific here.
Thank you very much. I think it's quite helpful. Thank you. And the last question, and then we will move to the biosimilars, because I want to give you enough time for the even more complex issue of the biosimilar. There is quite a number of questions that go around the therapeutic equivalence for a topical product. You've already answered for steroids, so maybe we can give them after the session some reference and guidance on this topic. But if you want to give, the question is, what kind of studies are needed to prove therapeutic equivalence for topical products, creams and gels?
Yes. Well, this is quite new territory in this time, because there is a draft guideline for evaluation of pharmaceutical equivalence for topical products. I'm not sure what's the exact name of the guideline. And this guideline helps to topical products in some way to avoid the need for therapeutic equivalence. And there is a description for extended pharmaceutical equivalence, which means that test and reference product are compared with respect to the quality. There are some in vitro release and permeation tests. There also might be useful to make a bioequivalence study, because for some topical products you can find also measurable plasma concentration or plasma levels of active substance, which does not correspond to the efficacy, but it can help as one of the requirements to describe the behavior of or to compare the behavior of a test and reference product. And if these quality and in vitro data are not applicable to make equivalence without the need of therapeutic equivalence study, then it should be again focused on the therapeutic indication of reference product. And for example, if it will be the non-steroidal anti-inflammatory drug to contusions of I don't know, of joints, or reducing of pain of some injuries of soft tissues, there should be equivalence where the people will use the test and reference and placebo, and they will record somehow the improvement of these medicines. In this therapeutic equivalence of topical products, there is one important thing, that there should be always the placebo arm, because for topical products there is a quite a high placebo response. And if you compare only test and reference product, these results are not reliable, because there is no information how, for example, for creams, how the placebo without the active substance can help. And without that, the data are usually inconclusive for the regulators, and the applications are rejected.
Thank you, Thomas. Yeah, the vehicle is quite important in terms of local action, and therefore the placebo effect is even reinforced by the vehicle. Okay, we will now move to the biosimilar. Thank you very much again for all the answers to the questions. We will send after the session some reference. I know there is another question, but we need to move to biosimilar because it's also a more complex topic and we need the time. Thank you.
Thank you. I'm happy that there are so many questions. I have a shorter part of my presentation focused on biosimilars, but these products are complex and quite difficult to compare with reference medicinal product. And I will start again with some definition. Biosimilar or biologically similar medicine is a biological medicine that has been developed to be similar to reference biological medicinal product. This is again abridged application like generics or hybrid applications for chemical substances. For biosimilars, there is Article 10.4 as an abridged application for biologics. Since this is quite a difficult field, I have here a reference to several guidelines. These guidelines describe how to prepare or how to evaluate the whole dossier, because this is not only about the bioequivalence study and the dissolution profiles. This usually needs to prove also efficacy and safety for biosimilar product. And apart from these general guidelines, which are mentioned on this slide, there is several specific guidelines for biologic medicinal products, which are sorted based on a type of active substance. So, for example, there is guideline for low molecular weight heparins, for interferon beta, and so on. There are more specific guidelines than I mentioned here, but you can all find them on EMA website. And all these groups of medicines have their specific requirements. So it's impossible to go through it in this presentation, but if you need anything about the specific requirements for biologic medicinal product from specific group, please go to EMA website, find this specific guideline, and you will find everything useful for this comparison and for these medicines. Probably you already saw these triangles, which describes how to characterize or how to describe the complexity of dossier for originator biologic medicines and also for biosimilar medicines. On the left side, you see the reversed triangle, which is for originators. Here is important the molecular characterization, but from the clinical point of view, the most important is the phase 2 and phase 3 clinical studies, which described and proved the efficacy and safety of the reference or originator biological product. On the right side, you can see the biosimilarity design, and here is quite clear that the comparability is based on quality data, on functional and physicochemical characterization of molecules and of the whole medicinal product. And from the clinical part, there is the great need for pharmacokinetic and pharmacodynamic behavioral or characterization and comparison between test and reference product. In some cases, even the efficacy clinical studies for biosimilar medicinal product are no longer needed, but usually there is a need at least for one study with also efficacy endpoint to compare similar behavior or similar efficacy between biosimilarity and originator medicinal product. And this should be my last slide. So what to say as a take-home message? The biosimilars have a complex chemical structure and are not considered identical to their reference medicinal product. I think you probably know that, but generics can be used as a generic substitution used by healthcare professionals. In case of one medicinal product is missing, the generic can be replaced by another generic, or the originator can be replaced by generic with expectation of the same efficacy in the same dose. And this is done automatically in pharmacies, for example, at least in Czech Republic. In case of biosimilars, we cannot say that these biosimilar is the same as the reference product. This substitution shouldn't occur without the decision of the physician and without the close monitoring of the efficacy of the biosimilar product newly administered to the patient. So the biosimilars cannot be substituted as a generics. This is important, and I think this is good to remember from this presentation. And that's all from my side. I thank you for your attention, and now I have prepared 3 questions to you.
Thank you, Thomas. Yes, we will do so the Slido. So please prepare yourself to answer in the Slido. We have already used it for the first session. You know that you can go to slido.com using the hashtag 1932 to enter this particular session. And please answer as many of you as possible the question that come now to the screen. What is the minimum required number of evaluable subjects in a bioequivalence study? Okay, the 12 in the chat is already an answer. Thank you for the answer. Anybody disagrees? More or less? You know, remember that in science, the majority is not always right. We need a few more responses. We have nearly 180 participants. And I know some of you are in a room with, and can probably only answer one response for everybody, but we need to go a little higher. So we wait a few more seconds or minutes, and then we will go to the answer. Okay, with 12 answer for a 13 answer now. Yes, good. Maybe, Thomas, you want to go to the discussion.
Yes, thank you. I prepared also the right answer, and you are correct. This is from the bioequivalence guideline, where is mentioned that the number of evaluable subjects in bioequivalence study should not be less than 12. Don't ask me why 12. I don't know. I never heard the reason why. Probably some statisticians decide that 12 is a good number, but I really don't know. But it's a correct number. So we can probably move to next question.
Yes, I think it's based on statistics, so the chances of finding a difference that is meaningful, but it's very few, actually. So the question is, for evaluation of bioequivalence in medicine product with high intra-subject variability, which parameter can be widened? Someone is still on the previous question. With a 12. But we have 2 responses. That's good. I like when we don't always agree. Do we go beyond 9 responses? 10, yes. So, Thomas, there seems to be a preference for the Cmax.
Yes, I think we can stay in Slido or it's fine. I again have a answer, and the Cmax is the correct answer. As you can see, highlighted in the yellow, the acceptance criteria can be widened for Cmax, I also had it on the slide, so I'm glad that at least 10 people listened my presentation. And here is the second yellow is mentioned that this does not apply for AUC. Okay, so we can move to the last question.
So the SmPC of paracetamol recommends administration with food to reduce possible gastrointestinal adverse events. Do you expect to see bioequivalence study under fed or fasted conditions? So you have two options. Please, we will see the response as they go on. Please vote. Thank you. If we could get a little more than 13 responses, that would be even better. Fantastic. We're now at 20 responses. Now, this question seems to have inspired everybody. Thomas?
Yeah, I'm happy.
Great. Went over 30. Thank you very much for your participation. Thomas, over to you.
Yes, thank you. I probably could say that if somebody thinks that neither of two questions is correct, that they could write it into chat. It was sketch question because the paracetamol is BCS class 1. Therefore, there is no need for bioequivalence study. However, in case of need to perform bioequivalence study, for example, in case of fixed dose combinations or different excipients, the correct answer is fasting, because the reason, as mentioned in a question, for to reduce gastrointestinal adverse events is the safety reason, and it's not a pharmacokinetic reason. Therefore, the most sensitive bioequivalence study is still under fasting conditions. This is not a very usual case, but sometimes it can happen that from pharmacokinetic reasons there is another recommendation than from the safety reasons. And in case of evaluation of equivalence between test and the reference product, it should always be taken into account a pharmacokinetics criterion, and the fasting condition is the most sensitive. Therefore, the bioequivalence study should be under fasting conditions.
Thank you very much, Thomas. Yeah, it's good to see, as I said before, that the majority is not always right. Okay. Do we have other questions from the audience? Free questions, clarification, discussion? You can either put it in the chat or take the floor if you want. So there is a question that are there some specific characteristics or differences of the bioequivalence study compared to generics? So yeah, what are the main difference between biosimilars and generics?
As I said, the biosimilars are much more complex, and it's much more difficult to perform all the tests and studies to compare with the reference product than in the generics. So the quality part of the documentation is much more extended in the biosimilars, and also there is I say in most cases of biosimilars, there is still a need to perform the efficacy study, at least small one, to prove the efficacy of biosimilar product. This is not applicable for generics. So generics are much more easier to compare with reference product than biosimilars.
Thank you. Yeah, because the manufacturing process has such an importance on the active substance for biosimilar. You cannot just measure what is in the blood. It has, and also some of these products don't act just through the plasma concentration. They also have interaction with receptors, which makes it very difficult to measure in terms of similar activity. Therefore, we have to have some clinical data as well. Just also a point, I think, for the reference product, as you said, for biosimilars, we now accept that the reference product is not sourced in the EU because of the need for human data. We don't want to repeat unnecessary studies and expose participants to a clinical trial when just for the sake of regulatory requirements. So the question is, are we going one day for products which are even simpler to compare to accept non-EU sourced reference product? That's an interesting question, I think, to be discussed with the Commission at some point in time. Do we have other questions or comments from the audience, please? Anybody wants to take the floor as a question verbally?
If you allow me, I would just like to make a comment about the substitution of the issue of substitution depends on the physician's prescription, but still, and I'm happy to hear in this presentation that the physician is the one who decides if a certain medicine will be substituted with biosimilar, because for sure it is not generic. It's similar to the reference product, but still there are cases in some countries. Unfortunately, at the moment, I don't have the right information which countries do practice this. They have a list of biosimilars that can be the originators that can be substituted with biosimilar, even in the pharmacy. These are rare cases, but are present in if we talk in global level. I think that in some European countries also, there are countries that not for all biosimilars, but they do have set a list of biosimilars that can substitute the reference one. Why I am talking about this? There are initiatives among different associations that ask the medicines agencies for the opinion if they would accept, if it is suitable to accept substitution, and we always go with the answer that it's the physician's decision, and we intend to go in that direction until we don't have maybe more experience on that that have been set for certain biosimilar that I mean it can be offered in a pharmacy instead of a reference product.
Thomas?
Yes. Yes. Thank you for this comment. Honestly, I'm not sure which countries in the Europe or in the world are willing to make a substitution of biological products in the pharmacy or without knowing the physician. Of course, probably in the beginning there was the intention to make let's say biological generics for these biological products to allow this substitution, but the praxis showed that it's not simply possible, and the people just can behave differently and just become decompensated based on this substitution of biosimilars. And I think, as you said, it's quite a new field, and we need more biosimilars and biological products and more time to evaluate or explore how the how these medicines will act in different people, and with more and more medicines, we will be definitely facing the situation that some medicine just expired or had some manufacturing problems, for example, and there are patients that were used to this biological medicine, and they are not able to use it anymore, and they should be switched to another biological, let's say biosimilar, or maybe back to originator. And there we will receive more and more information how it works with these changes in biologicals, and maybe in a few years or maybe more, we will have enough information how to work with this data, and if it will give us information that, for example, some certain biological molecules can be switched or maybe not. But currently, at least in Czech Republic, it's not recommended, and it's on a physician decision.
It's interesting, Thomas, because
Thank you very
actually,
much. I fully agree with you, Thomas.
can I intervene here? Because actually, the FDA has now accepted that the patient himself or herself can substitute in the pharmacy, deciding which product to a biosimilar to buy. I also think that with the time now that we have approved a number of biosimilar and we have the experience, the fact that they are interchangeable is exactly the whole purpose of the exercise. It's a bit contradictory to consider they are biosimilar and then to say, oh, but they are different. We are saying all the exercise about comparability and making sure that the results are the same in patients. So yes, some patients will have a difference. We also know that it exists with simple generics, but I think some of are also the fact that the patient don't like to change, and they will have a nocebo, if you want, a nocebo effect from the substitution. But it's not a simple question. At present in Europe, the situation is very different depending on the member state. They all have set up different ways of approaching substitution. It's not the business of EMA, but at the same time, as I said, we do a lot of exercise studies and demonstration of comparability to say at the end, we cannot tell you that they are interchangeable, which personally I find a bit contradictory. So I think with experience, we probably get more substitution. But the FDA has already moved away from no substitution or just the physician deciding on the substitution. So things are moving on. We have another question, maybe talking about possibility to classify a biological product as an auto-generic and not a biosimilar when the product remains the same with the same manufacturing process. And here I would like to say that auto-generic is not something that exists in our legislation. So I'm interested in knowing either it's an informed consent, and this is the same company producing the same product, but we do not have a concept of auto-generic in the EU legislation. But back to you, Thomas.
Well, this is not an easy question. I think that auto-generics are maybe set somehow in the national legislation of certain member states, but as far as I know, they are used or mentioned in case of chemical active substances or chemical medicinal products when the medicine is manufactured in the same facility, but the marketing authorization holder is different. In case of biological medicinal products, I'm afraid that even if the medicine is manufactured in the same facility, there will be some differences in the structure of active substance, because we are talking about such complex molecules, and if we sometimes compare different batches of biological products, there we can see some small differences. And therefore, I think that autogeneric is really not a good way how to proceed with biological products. But as Anya said, I think that it's not mentioned in a EU legislation.
No, I've never seen anything looking like autogeneric. But I understand the informed consent when a company producing a product give authorization to another marketing authorization holder to use the same product. Then that is what is there. And of course, in that case, you don't even need a bioequivalence because you just have to copy the dossier. But I guess there are some differences. I was not aware of this possibility. Any more questions or comments to Thomas? Thank you very much, Thomas. You've been bombarded with questions, which is very good. It's what we want, but you must be exhausted. I don't see any question in the chat or any raised hands so far. So it's last opportunities, please. And while we were waiting for the next question, I just wanted to say that we, as always, we plan to have an assessment of this, the whole training, and there will be a survey to be performed on the third day. So you have had the full, for 7 sessions, and we will ask you to fill it in between before the last session. So we have a full picture, because last time we got very few responses. We want each of you individually to answer the survey. So we have a good feeling of what did you like, what did you not like, what would you like to see. We already have some topics that have been identified by the contact points in the countries, but it's very important for us to know if you appreciate the format, the content, if you want more questions, and if you want more time for discussion. This is really important to make this session even better. We have 2 topics, just as an announcement. We have 2 topics that will come probably around June. One is on GMP, because that's a request you had, and the other one is on risk management plans. So remember that we have not finished. We continue with the training. It's remote. It's a bit disappointing for some of you who wanted to come to EMA. We'll be happy to welcome you, maybe all vaccinated next year. But in the meantime, at least the benefit is that we can engage with more people. And we had today about 180 people in your session, Thomas. So really a fantastic response. We want also more response of Slido, because again, if possible, most of you should answer, so we have a better idea of your understanding of what has been presented. So I still don't see any hands raised or questions. Last opportunity. If not, I would like to thank very much Thomas for this excellent presentation and all the questions and the answers, because that has been very, very interesting, and I think it really fulfill a need for the training. Thank you very much. Thank you also to my colleagues who are supporting technically, Clément and Sandra, who are on my left. And I think we will close the session here, and we will send you the presentation as promised. We will also send you some references, and I think there was one question that was not answered about the vet medicines, and we'll try to answer in writing. Thank you very much, and have a good lunch, because we meet again at 1:30 European time, 1:30. And for 1 hour and a half on COVID vaccines. I'm sure there will be also a lot of interest from all of you. Thank you very much, and talk to you soon. Bye.
Thank you very much. Bye-bye.
Hello, colleagues. Welcome back. I hope you had a nice lunch. lunch break for those who are still on the our time. We are going to have a very fascinating session now, webinar. And thank you, Manuela Mura, for presenting on vaccines for COVID-19 or against COVID-19. And I think you will all be interested in that presentation because it goes a little bit beyond the acquis communautaire, but at the same time, it's also a lesson on the way we approve vaccines and what are the elements we take into consideration. And so I give you the floor, Manuela. Just a reminder to the participant, mute yourself if you're not speaking, but don't hesitate to take the floor, raise your hand or put your questions in the chat. And Manuela, if you can introduce yourself and go on with your presentation. Thank you very much.
Thank you, Agnes. Good afternoon, everyone. My name is Manuela Mura, and I am a scientific officer in the Office of Biological Health Threats and Vaccines Strategy at the European Medicines Agency. And today I will tell you a little bit about the authorization of COVID-19 vaccines in the EU. You all know that the EMA is a decentralized body of the European Union, which is working on authorizing specific medicinal products by legislation since 1995, working together with a large network of regulatory authorities across the European Union member states, including 3 member states from the EEA. And all this network is reflected in 3 main scientific committees, which you will hear a lot about today. The CHMP, Committee for Human Medicinal Products, the PRAC, the Pharmacovigilance Risk Assessment Committee, and the PDCO, which is the Paediatric Committee. Just as a reminder, as I'm sure you may know this already, vaccines must be assessed by EMA via the centralized procedures. So that means not by the individual member states via the centralized procedures. If they are eligible under the mandatory scope, which means if they are produced by biotechnological means. If they are not biotech, the centralized procedure is still the preferential route, which is possible under the optional scopes, which are in the interest of patients or of the community. And this is an outline of what I will try to tell you about today. First of all, how EMA operates during a crisis and what is the COVID EMA task force or COVID ETF, which roles and responsibilities they have, and how EMA have contributed so far to approve COVID-19 vaccines faster. I will give you a brief overview of what we have authorized and what is still under evaluation and what are the requirements for approval, and an overview also of the dossier of the vaccines that have been approved so far. I will also give you a little overview of the pharmacovigilance monitoring, the EU pharmacovigilance plan, and finally, we will briefly touch upon the SARS-CoV-2 emerging variants and the need for variant vaccine and what we have for the seasonal influenza vaccines, what is the status for the strain change for these vaccines, and what can we use from this model to implement for COVID. So we will start with the first 3 bullet points. So during an health threat or a pandemic, EMA operates under the framework of a specific piece of legislation, which is the Decision 1082/2013, and has developed a plan, an internal guidance on which activities must be undertaken and role and responsibility of all the different EMA groups during a health threat. And within this framework, the EMA Pandemic Task Force of ETF is actually the key group, is a dedicated group since 2009. It is composed of experts from the regulatory European network and is responsible for the crisis-related regulatory activities, essential interaction with companies and data evaluation. The mandate of the ETF has been updated in 2020 to cope with the COVID-19 specificity, and therefore we speak about COVID ETF. It's chaired by EMA, and it includes representatives of the main scientific committees and working parties based on their expertise on vaccines means infectious disease trials, pediatric trials, but also quality of biological medicinal products. And recently has incorporated civil society representatives and representatives from the European Commission and the ECDC. The main roles and the responsibilities of the ETF are listed here. Essentially, the group is responsible for identifying promising candidates for COVID, review data emerging from the products in the pipeline, and engaging in discussion with companies with the aim to support development properly based on EMA requirements, and also based on an harmonized view at the global level. It is responsible for reviewing study protocols and to comment on clinical development via scientific advice, rapid or standard scientific advice, depending on the need of the company. And then once these products are submitted, they are also, the ETF is also responsible to contribute to product assessment by acting as peer reviewer and forum for discussions of the rolling review data assessment. And I will tell you more about this. Important bit is the second bullet point, the interaction with the CTFG, which is the Clinical Trial Facilitation Group, is a group responsible at the European level to approve clinical trials and to harmonize across Europe clinical trials conduct. And the interaction with ETF has been key during this pandemic. And it's something that will also improve in the future. It also contributes to the PRAC activities on emerging pharmacovigilance issues. And then there is also the big aspect of the public communication, the preparation and publishing of scientific positions and guidelines. And last but not least, interaction with international regulators and other stakeholders, which is a key aspect and has been extremely important during this pandemic to improve on the speed and to ensure that there is a concerted action in terms of regulatory decisions. Discussions are ongoing across Europe with the European Commission, the Health Security Committee, the European CDC, but internationally with the FDA, Health Canada, UK MHRA, Swissmedic, WHO, with or without the umbrella of the ICMRA, which is the larger, the hub for international collaboration, including global regulatory authorities. EMA has greatly contributed to faster approval of COVID-19 vaccines in several ways. Early on during the pandemic, we have developed a tool like an inventory of all the tools that we have and that can be improved and adapted to COVID to support development and to support evaluation. At the level of development, we have the rapid scientific advice, which means that the process has been shortened from 70 to 20 days. And also, we held informal teleconferences with developers to brainstorm on any aspect of development or submission. And also, the Paediatric Investigational Plan is endorsed by PDCO in a rapid fashion, shortened from 120 to 20 days. In terms of evaluation to support, the rolling review is one of the key tools that we have used and will continue to use. The COVID ETF is responsible to decide when the rapporteurs are appointed for a product, when the rolling review can start based on the level of evidence available, and also then to support the CHMP in the review of the data, as I said earlier. When the rolling review is not available, or especially for repurposed products, which are products that are already authorized, but that they can be developed against COVID-2, therefore an extension of indication, an accelerated assessment is still a good tool in this case to use, which can shorten for example, for a marketing authorization, the assessment down to 150 days. And finally, we have other tools in collaboration with the member states, which are the compassionate use or emergency use authorization. These type of tools are applicable only to products which are not yet authorized, and I will tell you more in a couple of slides. Marketing authorization, as I said, based on the experience from the 2009 influenza pandemic, the rolling review has been key in accelerated evaluation, whilst still ensuring a robust scientific opinion. And the key of this procedure is that the company is able to submit formally, because it's actually a formal procedure, to submit data as the data has been developed and is becoming available directly to the AMA for assessment without waiting that the full dossier is ready for submission as it would normally happen. And only when a sufficient amount of data is gathered, is accumulated, that can allow for a conclusion on a benefit risk, can the applicant submit a proper formal conditional marketing authorization, or anyway a marketing authorization application, which implies that then also the pediatric investigational plan will have to be finalized and agreed with PDCO, and then the procedure can be concluded. Other aspects that are accelerated are the fact that, for example, opinion and assessment reports can be adopted via written procedure without having a plenary committee ongoing. In particular, this is very helpful in case of the rolling review, because we can have 4 or 5 rolling review waves, and therefore we have as many interim opinions. Also, translation checks are done already during the rolling review. The Commission decision is able to the European Commission is able to grant a Commission decision in a matter of days compared to the 67 days normally required. And finally, we are aiming to publish an EPAR, which is a European Public Assessment Report. That is all the documents supporting the CHMP conclusion on an authorization on a product immediately following the Commission decision. In terms of conditional marketing authorization, this is what I mentioned. You may have heard of this already, but just to give you a reminder, this is an important tool during an emergency, as one of the scope as foreseen by the legislation is that this is a tool that can be used during emergency situation. Otherwise, the conditional marketing authorization can be used. Sure, I will try to talk slower, sorry. A conditional marketing authorization can also be used for life-threatening diseases or seriously debilitating diseases, or for orphan medicinal products. But there are some criteria which are important to be met for a CMA to be allowed and to move forward. The concept is that a conditional marketing authorization is based on less comprehensive data than a normal marketing authorization. But there are specific obligations that the marketing authorization holder will have to complete post-authorization. In addition to this, even if less comprehensive, the data must be sufficient for the risk-benefit balance to be positive. And the applicant will be in a position to provide comprehensive clinical data, that would be the specific obligation, within predefined timelines by law. The product must fulfill an unmet medical need, and the benefit to public health for the immediate availability on the market of the product concerned are far larger than the risk inherent to the fact that the data are not comprehensive compared to a normal marketing authorization. The CHMP, once the specific obligation will be submitted, so once the full set of data imposed to the company will be submitted, will review this data and will decide when these obligations are actually fulfilled adequately, so that the conditional marketing authorization can be transformed into a full marketing authorization. And a few words also on the compassionate use. As I said, this is a tool which allows member states to use a product that is not yet authorized and is still under clinical trials, so it has to be in advanced stages of development. A compassionate use, as per a legal definition set by Article 83 of Regulation 726 of 2004, is actually limited to new treatment. But for vaccines, we have a similar tool, which can be extrapolated from Article 5.2 of Directive 2001/83, that allows member state to temporarily use an unauthorized medicine in case of an emerging health threat. And this is what the legal provision that is used for vaccine and has been used by some of the member states even during this pandemic, even though most of them have agreed commonly to wait for the conditional marketing authorization by EMA before using any vaccine. The compassionate use or emergency use authorization in the European Union is a competence of the member state, is within under their remit, is within under their legal provision. So there are also slight differences how this is implemented across the member states. Only a member state can initiate the request for a compassionate use, but what the EMA can do is because it's informed by a member state whenever they want to use this tool, they can provide a recommendation through the CHMP for scientific aspects of the emergency use, so that there is an harmonization and a common approach across the member states that wish to use this tool. And finally, we have Article 58 of Regulation 726. This is known as Article 58 products. And basically, this piece of legislation is giving the EMA the possibility, working in collaboration with the WHO, to generate a scientific opinion sent directly to WHO to support the use of a product outside of the European Union. This hasn't been used so far simply because it could be faster for WHO to use a pre-qualification procedure based on the existing opinions and authorization by stringent regulatory authorities such as EMA. However, is a tool that is in our apparatus that it can be used. And this is just a slide to show you the importance of these regulatory tools to support a faster regulatory authorization. On the top, you see what is normally happening in terms of clinical trial, preclinical studies, which can take up to 4 years, clinical trial application or IND, depending on the regulatory region in the world, and then the clinical trial development, phase 2 and phase 3, each of which can take 2 years or longer. And only after all these data are gathered together in a dossier as per regulatory requirements can a regulatory authority like FDA or EMA start the review, which can take up to 2 years, and is issuing a full marketing authorization. And this is followed by large-scale production and distribution of a vaccine. And overall, this process can take up to 15 years or longer. So what happened during the pandemic, as you well know, there has been first of all, a reduction in the time required to finalize a development, both in terms of looking at what data we already have, for example, from SARS or MERS, what can be used, what can be extrapolated, streamlining on clinical development, but also dedicating more resources, more money, especially in terms of clinical trials and adaptive approaches, adaptive study design, so that the different phases can actually overlap. Everything is compressed. There is no cutting corners, but everything can be compressed in a very short time frame. And the key aspect in red, circled in red here, is that while we wait until the end to start our review, normally, in this case, we are able to initiate the rolling review already during phase 1. As soon as we have some data showing proof of concept in terms of immunogenicity and acceptable reactogenicity and tolerability of a vaccine, we can initiate the rolling review assessment as the trials are still ongoing. And then a conditional marketing authorization can be submitted as soon as we have some efficacy data, interim analysis data from the phase 3 pivotal study. This allows to reach a conditional marketing authorization in a matter of few months, so that overall this process, instead of the 15 years, can take up to 10 months or anyway less than 2 years. Obviously, trials must continue.
Maybe, Manuela, can we make a little break here to try to see if there are questions, because it goes quite fast and I think it's a lot of information, so maybe checking with the
Sure.
I think we have a question on what is available after the session. Yeah, the session is recorded, so there will be a distribution of the recording afterwards. But maybe some question from the audience on understanding the difference between rolling review and normal marketing authorization. This morning we had, for an orphan drug, also a case of CMA, so that was quite good because it has allowed to have the same concept explained already. So you just repeated it, which is good for training. Yeah, maybe
I think we have some questions prepared. Maybe if Clemence can put them up. I think at this stage we have some questions prepared.
Okay, thank you. So we will go to Slido. So please prepare your computer or your smartphone to go to Slido and to answer the question. Remember that it's slido.com with the hashtag 1932 to enter the room. And now you have the question. So maybe you can start answering. And we hope to see many, many responses. Like last time, we went to more than 30. That was good. Let's beat this record. Thank you. All of the above question refers to the three options, other options, of course. Yeah, we definitely beat the previous one. That's very good. Thank you, Manuela. Maybe you can explain.
Yes, it's very good. The first is the right answer. I mean, the other points are also partially correct, but it's just a small part of the role of the COVID ETF, which has a key role in the regulatory activities during a health threat crisis, especially interacting with companies and evaluating the data of the pandemic products as they become available via rolling review or other procedures. So we can move to the other question. Yeah, what tools has EMA put in place to contribute to faster approval of vaccines in the EU? Also, in this case, all of the above is referring to the other questions. And I can see that we have a very large number of responses here as well. And here the correct response is all of the above. The rest are obviously the other two are correct, but we both use tools during development as well as during assessment, because supporting companies during scientific advice and during PIPA evaluation is key, because that means that the development is conducted as best as possible based on the requirements of the EMA. And even though the response of the CHMP to a scientific advice is not legally binding, still it was shown that if company adheres to a scientific advice, they have higher chances to reach a positive opinion. So it is extremely important to have this type of discussions with company, and especially a pediatric investigational plan can represent a bottleneck. If company companies are preparing this and submitting this too late. Normally, they should start already at the time of the phase 1 study to look into a pediatric investigational plan. This is very important to avoid to wait at the last moment, which can then, if there is no agreement reached with the pediatric committee, it's actually a showstopper for a submission of a marketing authorization, because a compliance check based on the pediatric studies is a mandatory requirement at the time of validation of a conditional marketing authorization, or any marketing authorization for that matter. And I think we have a final question for this session, which is, what is a rolling review?
Thank you. Now we have gone very high, so that also shows the interest. We're nearly at 50 responses, which is excellent.
Okay, we are also over 40 responses and growing. Yeah, and I think 96% of people got it right. It's a special procedure which is used during health emergencies to accelerate as much as possible the EU approval of medicines, including vaccines. So it can be used for any medicines. And I had questions in the past whether, if this is so successful, why don't we use it even outside of an emergency? And I have to say that it requires a very dedicated group, resources at the level of the EMA and the whole European network. So that it's so shortened and compressed and intense that a lot of people are working to support it in a very short time frame, and therefore it really is something that can be used only during an emergency. So it is not a type of scientific advice. A rolling review can only occur when a set of data is, when an overall amount of data is present, that the COVID ETF can decide, okay, we have enough, that could be used for a benefit risk conclusion, and the company can start to submit bit by bit in waves of submissions. Could be, for example, the non-clinical data first, followed by the manufacturing data, followed by part of the clinical data as it becomes available in the duration of several months. So it happens after basically a scientific advice phase. Even during a rolling review, scientific advice can still be requested, but this needs to be carefully timed by the applicant. Yeah, and that's all. I think we can proceed with the presentation. That was the last question for this session.
Yes, please.
Okay, so I will now give you a short overview of which vaccines are authorized under scientific advice or under evaluation in the next couple of slides. And this is just, I will not speak a lot about this. You will be able to read it at your leisure afterwards, if you are inclined to do so. It includes a link to our webpage of a table that lists all the products which have interacted with the COVID ETF and EMA, informally or formally. And also here is a breakdown of products that have received scientific advice based on platform. So as you can see, very extensive, as also the scientific advice, it's free. So a lot of even small companies were able to access this tool. And here is just a slide to give you a little bit of content in terms of what we discussed with companies, across companies. For example, for clinical topics, we discussed essentially phase 3 efficacy study design, from the success criteria to the endpoints, to the possibility to have interim analysis or pooling of data to reach a conclusion on a benefit risk. The duration of safety follow-up was also an important part of the discussion in the past month. Wording of the indication, aspects of pediatric development, inclusion of people with comorbidities and of elder age in the pivotal studies, and pharmacovigilance and post-authorization studies also for effectiveness, and so on and so forth. And here you also see a summary of topics for the non-clinical developments, especially, for example, looking at endpoints for enhancement of disease or species for challenge studies, etc., etc. Now, which vaccines are authorized? You know very well. Janssen was the last one in March, and then AstraZeneca, Comirnaty from BioNTech, Pfizer, and Moderna were authorized in the previous month. And ongoing, we have the rolling reviews for Novavax, which started in February, as well as CureVac and other messenger RNA. And finally, also the Gamaleya vaccine, Sputnik V, started in March. This is the last one. And a few more are coming up as well. Now, I will touch also a little bit about requirements for vaccine approval, and I will give you an overview of the clinical development plans for, well, actually the dossier of the authorized vaccines before breaking for the next set of questions. Now, in November, the COVID ETF published a guidance for developers, and including a Q&A for the general public, to describe the main criteria that were expected for phase 3 efficacy trial, the pivotal trial for approving a COVID-19 vaccine. Essentially, at least one large-scale study is required and should be designed as standard prevention trial with the target population as per indication included, and a primary analysis focusing on subject seronegatives at baseline. The primary endpoint, as you can see in the second bullet point, is prevention against symptomatic COVID-19 disease of any severity. This is considered an acceptable primary endpoint. And secondary endpoints can relate to prevention of a severe disease or prevention of infection or hospitalization, etc. Now, the studies, they don't have to be powered for subgroups, for example, age or comorbidity. However, inclusion of elderly is encouraged if the posology is adequate, obviously. At least 25% is considered an acceptable number. People with comorbidities also are strongly encouraged to be included, because elderly and comorbidities are the individuals who are more at risk for severe COVID-19 and death, as well as ethnic minorities. So that the trial population is representative as much as possible of the population that will be then the target for vaccination. It is not possible, in the view of the COVID ETF, to define a precise criteria for approval of a vaccine, for a vaccine to be defined successful. However, it is recognized that a study to support approval must be adequately powered, which is, for example, based on an assumption that the lower bound of the 95% confidence interval should be at least 20, better 30%, and vaccine efficacy estimate should be around 50-60%. This is considered a well-powered studies of an adequately sample size. For submission of a conditional marketing authorization, there are specific criteria which must be met. In terms of efficacy interim analysis, a successful analysis, for example, is based on a lower bound of the 95% confidence interval above 20-30% with a point estimate of, for example, 70%. This could support an early decision, even though it's not a final analysis, but a conditional or emergency use authorization could be based on this type of data, and was based on this type of data for many of the vaccines. And in terms of safety, at least 3,000 subjects followed up for at least 4-6 weeks is required for a conditional marketing authorization. Obviously, the data set for safety is expected to be much larger than that because of the need to have efficacy demonstrated in a very short time frame, which means that the sample size must be large to be able to detect enough cases of COVID in the placebo group fast enough. However, we won't be able to, I mean, normally we would wait 2 or 3 years, depending on the duration of the trial, for an authorization. In this case, we define this time of follow-up for 6 weeks as the minimum data set for a conditional marketing authorization based on the time window for a plausible relation of adverse events with vaccination. Long-term follow-up is essential for both safety and efficacy. Therefore, EMA is always strongly recommended that studies continue after reaching the primary endpoint for efficacy to the end of the study, which is not, therefore not, overall for 6 weeks, but it goes up to 2 years, normally 1-2 years. This is the duration of the studies, which is able then to look into more rare adverse events or into what happens with efficacy and immunogenicity once antibody decay, how they decay, the kinetics of antibodies, and also in terms of potential occurrence of enhancement of disease once the antibodies are disappearing. With this data set, we will be able to detect uncommon side effects. Rare adverse reaction, that is with the frequency less than 1 into 1,000, are really unlikely to be detected in a normal clinical trial pre-approval. And this is normal for any vaccine. And even in this case, despite the large data set that we have seen with some of the COVID-19 vaccines, which were unforeseen before. But still, we require post-authorization surveillance studies and safety study as needed to be able to investigate or detect rare adverse events. And finally, important also to define long-term protection and immunogenicity, especially in view of determining when a booster dose must be given, and if and when a booster dose is to be given. And this is just an overview of the dossier of the 4 approved COVID-19 vaccines so far. Here is 4 non-clinical. And just you will be able to go into this.
Sorry to interrupt again. I think because we have only 20 minutes left, I suggest that you summarize a little bit the messages and we can have a bit of time for discussion. Thank you.
Absolutely. So here just we have pharmacodynamics immunogenicity studies and efficacy, that is challenge studies, normally required, and then toxicology studies with biodistribution and DART study of development studies for non-clinical. And here, if you look at the clinical, you can see a summary of the phase 1-2 studies, or more likely the studies are embedded into the pivotal phase 3 in an adaptive design. And you also have a summary of the efficacy results from the primary endpoint in seronegatives, normally determined after 2 doses, but for Janssen only after 1 dose, which is how actually it was authorized. And then the only difference is that AstraZeneca was based on pooled data set, and they had limited data in elderly because they had a larger studies ongoing in the US, which is actually coming up. And here you can also see the data from efficacy against hospitalized and severe COVID for your perusal. And this is just an overview of the pharmacovigilance data coming up for the vaccines used so far in Europe. Here you see the exposure in terms of millions of doses, and the suspected ADRs based on ultravigilance are actually, you see, depending on exposure, correlated with exposure. You can see the identified risks so far, which are very similar, and what were under PRAC assessment as of 22 March when I prepared this presentation, but you know very well that now we have some outcomes from the PRAC assessment already for some of these vaccines. And I think at this stage I have a few slides with questions from what I said, but obviously whilst they are putting up the Slido, I think I can take any other questions from you.
Thank you, and Manuela. Let me see. I don't think we have questions in the chat, but maybe someone wants to take the floor and ask a question directly. Just unmute yourself and join the discussion. No, but then we go to Slido. I think people feel probably more comfortable and less shy. Thank you.
as number continue to grow, I can already start to say that this. There is not, in principle, there is not a correct answer, but in terms of what EMA decided in Europe, the correct answer is the number 1, which is what most of the people correctly ticked. It is not possible in the view of the COVID ETF to define a threshold for efficacy for approval. It is important that trials are adequately powered based on the lower bound of the 95% confidence interval, and that ensures that data are sufficiently robust to define a benefit risk. But we were not able to define, okay, a vaccine must be 50% efficacious for approval, because if a vaccine is 48% efficacious, then we would not be able to be in a position not to approve such vaccine if that would be the only vaccine available, for example. So this is a very, at the beginning, when this guidance was established in November, it was a very delicate matter, but it was felt important starting on the basis of a very robust clinical trial that we are able to define based on the overall evidence when a vaccine is efficacious and when a positive risk is a benefit risk balance is positive without setting a specific threshold.
Manuela, maybe what
yes.
could be interesting is also to say that when we got the two first vaccine, which was the mRNA vaccine, Moderna and Pfizer BioNTech, we were happily surprised by the level of efficacy that we saw, because we were in the 90+%, rather than what we feared we would have, which was this sort of not very satisfactory, but better than nothing. We started from nothing. Now we're in a different situation because we are becoming quite picky, saying if we have that level of efficacy, how do we cope with vaccines that are of a lower level?
Yeah, and you are totally right, Agnes, and that is probably what is, what NITAGs and Minister of Health across member states are scratching their head around in terms of vaccination campaign and prioritizing vaccines and risk groups for vaccination. In terms of EMA, we will approve any vaccine that is considered having a positive benefit risk balance based on the overall evidence available. And in terms of comparability, this is not what we would be, this is not in our remit, but definitely, Agnes, you're right. That was a totally different mindset back in November before seeing any of the efficacy data that then came up.
Yeah,
we can move to the next question.
The point that the fact that we have seen different results for different vaccines doesn't mean that they are so different. It's just that the way they were measured were completely independent, not necessarily using the same test, not the same population. So we should be very careful about comparison of efficacy through just parallel results. That's what I also wanted to highlight.
Correct.
Thank you.
Correct. Yes, of course. They have done different trials, different population, different assay, and therefore it is very difficult to compare immunogenicity result or efficacy result. That is not something that is very easy to do or should be done to be correct from a scientific perspective. Okay, I think we have another question.
Yeah, and we have a question in the chat. Is it possible to see the overview again for the efficacy of all approved vaccines? So I think maybe we can go back after to your slide in the presentation, but let's do the Slido first and then go back to the presentation you made. So all participants, be brave, take a risk, answer the question. It's not necessarily a wrong answer. It's more intended to trigger a discussion. So don't hesitate to answer.
Yes, very good. We passed again the threshold of 30. So then, I think in the interest of time, 88% were actually correct. It is not necessary it is not required for a study to be powered per subgroup. This would affect feasibility, sample size. We would need to wait much longer, and it is not ultimately required because efficacy can be anyway extrapolated based on the trends that can be seen with an adequate proportion of elderly and comorbidities included. So basically, we are looking at a signal here. I think we can move to the other question.
We will go to the question on the overview of efficacy first, and then we also have a question in the chat, another question in the chat.
Yes, so I think the question in the chat, is it, no, how to be evaluated safety of messages such as tumorigenicity. So I will start with a question from Luca Santi. Is the rolling review applicable to a new marketing authorization or to post-approval variation as well? In principle, it could be applicable to both. The point is that a variation post-approval, for example, an extension of indication, for a rolling review to make sense must be a large data set. If it's only like normally happens for an extension, one clinical trial, it doesn't make any sense to have an assessment in bits and pieces because the trial is one. And also from an assessment perspective, it would be very difficult to conclude on anything if we don't see the whole trial. And therefore, factually, the rolling review is really best placed for a marketing authorization rather than an individual variation. And then another question on the safety of the messenger RNA vaccines in terms of tumorigenicity, fertility, and long-time risk reaction. So normally carcinogenicity assays are not required for a vaccine. For the messenger RNA vaccines, because they carried some new excipients in terms of lipid nanoparticles embedded in the construct. Some genotoxicity assay were performed, and that was considered sufficient to eliminate the potential for a genotoxicity. And in terms of fertility, well, that was evaluated in the DART studies in the context of the toxicology studies as well, and there were no issues foreseen. And in terms of long time, which I assume means long-term safety data, this is still being collected during pivotal clinical trials as we speak and will be submitted for assessment in addition to the ongoing pharmacovigilance, which I will touch upon later.
Maybe one point to insist on, Manuela, the fact that the mRNA vaccine does not insert any genetic code into the body. It is actually eliminated. It is just helps producing the protein, the spike protein, but it is not incorporated in the genetic in the body. So there is no fear of producing for long term some of the elements which I think are fueling the fear of long-term effect of that type of vaccine.
Correct. Yes, fully correct.
Okay, maybe we can move on because I think time is running. Thank you very much.
Yes, and just a brief overview of the pharmacovigilance monitoring. We have a preparedness plan for monitoring that is published on our website. Based on the lesson learned from the H1N1 influenza pandemic, and looking at three pillars: signal detection methods, which are key for safety monitoring, looking at new methodology, prioritization and assessment of the signals, and then collaboration with academia, research centers, and international regulators for exchanging information and data on vulnerable population, active data collection on rare risks. And I'm talking about ICMRA, for example, but also trying to discuss pregnancy studies or setting up registries or international cohorts across states or international, which could help in a faster detection of issues. And finally, the other pillar is transparency and communication, which are key. As we know, safety and pharmacovigilance are very important from a public perception, and therefore to communicate timely and clearly on why decisions are taken and based on what data is very important for public, patients, and also healthcare professionals to ensure that the vaccination campaign is not compromised by misunderstandings. Therefore, communication is enhanced, and in terms of transparency, we publish documents which we did not used to, for example, the full RMP on our website. And here are 3 slides on the monitoring post-approval. Just to briefly tell you that overall, whatever is in place for other vaccines, the GVP modules are also applicable, of course, for COVID. They are setting up the basis for the pharmacovigilance. In addition to that, a core RMP for the COVID-19 vaccines has been developed to guide developers in the different parts that the RMP should include, with specificities for COVID when available, when applicable, and this is published on our website. And in terms of surveillance, PSURs are normally submitted every 6 months in the first year, and then their frequency decreases after. But in addition, monthly summary safety reports are published, are actually submitted by the company, assessed, and some of our analyses are also published, and including exposure data and information on ADRs and adverse event of special interest. It is key that member states are gathering exposure data in a timely manner so that the adverse events can be contextualized to the use by using registers, and this is also able to enables the EMA to perform pharmacovigilance analysis such as observed versus expected analysis. And for this to happen, batch and product traceability is key by use, for example, of stickers. These measures are defined in the RMP, and they are implemented in agreement with the member states. Spontaneous reporting of suspected ADRs should occur as mentioned in the GVP modules. And in addition, there is some guidance available for the individual case safety reports based on COVID-19 or specific MEDRA terms for reporting. It is very important that the quality and the completeness of the reports is taken care of, because that would enable correct causality assessment as much as possible, and also that the individual case safety reports are timely submitted to the reporting authority less than 15 days for adverse event of special interest or life-threatening reactions, for example, is what is recommended. In addition to the normal monitoring, we have, or enhanced monitoring, we have a large part of the post-approval that pharmacovigilance that is dedicated to enhanced to research. Observational research in terms of safety studies imposed to the MAH or proposed by the MAH are included in the RMP to further characterize the risks identified or potential, or to investigate missing information. For example, in pregnancy or in other vulnerable population. And in addition, EMA has contracted academia and private partner to support the network with dedicated initiatives, such as the Access Project in collaboration with the Utrecht University, to look at coverage data and especially safety and effectiveness study protocols, to develop these protocols, but especially to establish background rates of adverse event of special interest, which are essential then to do observed versus expected analysis, for example. The Consign project looking at the impact of COVID-19 disease on pregnancy to support decision on vaccination, and the early safety monitoring studies, which is will start or started already in this year, to complement the spontaneous reporting system on ADRs. In addition, there is a large initiative funded by the European Commission to support the conduct of observational safety, but also effectiveness studies, extremely important, as part of a joint action between the ECDC and the EMA. So it's a full-blown vaccine monitoring program, which will run into 2022, looking at safety and effectiveness, and is going to be very important and is under discussion at this stage. And then just the last slide on signal management that has been actually accelerated, prioritized based on principle. The timelines have been expedited so that, as happened for the AstraZeneca, we can reach a decision very, very quickly. Exchange of information at the level of the EU is also key. You can see here all the tools that we use, but also in collaboration with international partners. And as I mentioned, communication and transparency also very important. And I think I have here one question, if we want to use the Slido or if you have...
I see no question in the chat, so maybe let's go for the Slido and then we will have, unfortunately, to conclude. But if you have questions in the chat, please put them in the chat, because then if we do not have the time to answer verbally, we can also provide some answers afterwards. So thank you.
Yes, and while people are responding to the Slido, I have a question from Paolo Porcelli on which safety analyses have been published, and I will look into that and I will send you a link later on.
You can send your question to everybody, because it may also remind everyone of some points, and so don't hesitate to include your question in the... I know I'm asking you to do two things, answer Slido and write in the chat, but I'm sure you can do both at the same time.
Yes, and here we are over 30 participants, so they are all correct. It's both B and C, improved and accelerated signal detection methods, and international and research center collaboration and increased transparency and communication are the key pillars of the pharmacovigilance monitoring for COVID vaccines. So the existing process are the basis to start with, but they are not essential. They've been fine-tuned and enhanced for COVID-19 pandemic. And I think we can move back to the presentation unless there are questions from participants. You can interrupt me anytime. And here is the last bit of my presentation on the variants and the need for vaccine and what can we learn from influenza. You know that the variants are an increasing problem. Here there are the three main variants of concern at the moment: the B117, which is the so-called Kent variant, the B1351, which is a South African variant, and the P1, which is the Brazilian variant, just based on where they were detected first. You know that WHO is looking into detecting variants in a regular and continuous basis, and many variants are arising and is normal with this type of virus, but some of them will be defined as of interest or of concern depending on whether they are able to transmit and then take over the existing strain, and then they are able to alter the phenotypic aspect of the current strain. That is, they are able to increase transmissibility or to increase severity. So definitely all of these three seem to be able to increase transmissibility, and they have starting to take over in certain regions the existing virus, and possibly the South African and the Brazilians are also involved into some improved severity of disease. And in this context, it's important to look at the data from all the authorized vaccine to understand whether they are able to cross-neutralize a new variant, or whether this is not happening, and therefore we need to consider whether it's necessary to change the composition of the vaccine. And here is some data which are becoming old as we speak, because new data came last week as well. But just to show you in an indicative way that, for example, this is from Moderna, but similar data were available from Comirnaty, that in the top panel, neutralizing antibodies raised by the existing vaccine, the parent vaccine, are able to neutralize also the Kent variant more or less in the same way. However, this is not the same for the South African variant, where there is a drop in the bottom panels of six-fold in terms of cross-neutralization. And this we don't know exactly how much implication has in terms of efficacy, considering that these vaccines are extremely efficacious, probably very limited, and we could definitely expect that at least a protection against severe COVID is maintained, but this is something that will need to be verified. And this is a piece of efficacy data that you are well aware of from the AstraZeneca vaccine, showing no efficacy against the South African variant in a very small study, which will have to then be also confirmed. So it's important that we look at all these data as they are generated to understand what needs to be done. And this is a discussion that is happening also at the level of WHO and related stakeholders. This is what happens for the annual strain change for influenza vaccines, starting from February each year until June, when the companies are submitting their procedure to the relevant competent authorities. There are a number of steps which are required in order to understand whether a specific variant needs to be included in the composition for the following years. And then this new virus needs to be produced by the company, standardized using the reagents, and then the variation needs to be submitted to the EMA. And this is an overview of the variation that is done by the EMA to change composition of the seasonal influenza vaccine in a matter of 55 days maximum. Based on law, we are able to have an opinion approved on the new variant each year for influenza vaccines. The possibility to change the composition of an influenza vaccine via variation is specified in the legislation. You see here on the right-hand side the reference. And so you see, for example, Article 18 and 21 of Regulation 1234 of 2008 is allowing the possibility for seasonal and pandemic, respectively, vaccine to be varied via type 2 post-authorization rather than having a new marketing authorization as it would happen when you change composition active substance for any other product. In addition, Article 21 also allows for less comprehensive data for a pandemic vaccine to be submitted and for shorter timelines for assessment. For some veterinary vaccines, it's very interesting to note, for example, flu, but also foot and mouth disease or blue tongue, that the derogation is actually broader. They are able not only to include, to replace the strain with another one, but also to add a new strain. So creating what is called a multi-strain vaccine. And these are all interesting aspects which could be considered for COVID. Now, in terms of influenza still, we had a change in the guideline in 2017, and this touched upon the requirement for the annual strain change for seasonal influenza vaccines. For example, before there were the requirement to conduct a small confirmatory clinical trial for reactogenicity, tolerability, and immunogenicity, but this is no longer required because based on decades of experience, we're not getting much more information from this trial than what is already known from the initial marketing authorization, and therefore, at this day, only quality and manufacturing data is necessary to change variation for an influenza vaccine. For a pandemic vaccine, variation is also similar, but in addition, we also we consider it preferable that the company manages to gather some clinical data, at least in adults and possibly in elderly as well, but depending on the emergency of the situation. So this is not a strict requirement. Obviously, EMA considers post-authorization continuous monitoring of vaccines performance essential over the years, which means we are looking at yearly effectiveness studies and yearly reports of enhanced safety surveillance to understand that despite the change in composition, the vaccine continues to behave as it is expected in terms of reactogenicity in the population indicated, to confirm tolerability of the new strain, and to rule out issues that could be, for example, on quality aspects, but they are impacting effectiveness of the vaccine. For example, like it happened for the MedImmune live attenuated nasal influenza vaccine, which had some issue in the H1N1 component, which led to a loss of effectiveness. So this is very key that this monitoring is done. So what of this can be applied to COVID-19 vaccine? The WHO processes and tools to support regulators in their decisions related to strain changes to influenza vaccine provide a potential model for COVID-19 vaccine, but needs adaptation because clearly there are some differences in how the viruses are behaving and what can be done for COVID. In terms of strain replacement or addition to an authorized parent vaccine, normally, because COVID vaccines are not included in the legislation as flu are, normally this would trigger the requirement for a new marketing authorization based on current provision. However, it is definitely much faster to do what we are doing for influenza vaccines, so to vary the terms of the parent vaccine marketing authorization or to create a multi-strain umbrella marketing authorization as for vet vaccine, but this requires a change in the legislation and the European Commission is looking into this as we speak, so we will hear more. But also the COVID ETF published a guidance at the end of February on the regulatory requirements for variant vaccines. This is in terms of scientific requirements. And essentially, these guidance apply to vaccines that are already authorized and for which a variant is needed. So in this situation, the strain change should be supported by quality, immunogenicity, and safety data. But this situation is acceptable only for vaccines that have, as I said, they're authorized, but also the variant is based on the originator platform. So the manufacturing is basically exactly the same, except that we are switching the antigen. Because in this case, we could use a streamlined quality dossier, absence of non-clinical data. We don't believe it is necessary. And in terms of efficacy, this can be based on an immunobridging strategy whereby the new variant is able to demonstrate non-inferiority of responses versus the authorized parent vaccine for which efficacy was demonstrated. And for this to be proven, they have to find a seronegative population that didn't have COVID before or was not vaccinated before. A variant could also be used as a booster in people who were previously vaccinated, and the guidance is also foreseeing this aspect. But as for influenza vaccine, also for COVID, surely continuous monitoring post-authorization is essential by enhanced safety surveillance or effectiveness studies to make sure that the vaccine, especially in case we don't have efficacy data, continues to behave as it is expected. And before the summary, I think I have also a couple of questions with Slido. But as before, you can raise questions. Yeah, so I have a question. In the case of COVID variant vaccine, which kind of clinical data requested? So safety are requested as well, but we are not requiring a large database or 30,000 or 40,000 people, because so far we are looking into the possibility of a variant vaccine for an authorized vaccine. So we have very large safety data set to start with. Therefore, only a smaller sample size looking at reactogenicity with the new variant is necessary. And in terms of efficacy, immunobridging, so based on immunogenicity data, then of course it would be easier with the correlator protection. A correlator protection is not there yet, and therefore in this case we would need to immunobridge with versus the authorized vaccine for which efficacy was demonstrated. Because if we had any correlator protection, we would simply need to look at whether the vaccine is able to induce antibodies, neutralizing antibodies above a defined threshold that is correlated with protection.
Manuela, maybe I can go on this because it's a very important point, this correlates of protection we've been discussing every time, say, oh, we don't have them. What it means for this particular vaccine, if I understand well, I'm not a specialist, is that we would need to say neutralizing antibody are the way the vaccine protects against COVID. If you reach a certain level of neutralizing antibody, you expect the vaccines to be effective. But in fact, what we know is that it's not just neutralizing antibodies. There is also an element of cellular immunity, which is far more difficult to measure, especially quantitatively, and requires tests that are far more complex to manage. Am I right, or is there something I'm missing?
No, you are right. You are definitely right. There are large initiatives, especially driven by WHO, looking into this. And more there is evidence coming up that neutralizing antibodies seem to be doing the trick, so seem to be the best immune marker that can correlate with efficacy. But we're not there yet, and anyway, to have an established correlate in regulatory terms, they will have, individual companies will have to provide sufficient data to show that whether this is a surrogate or is a proper correlate. So there is a lot of studies and a lot of data that needs to be generated. And obviously, we don't have the full answer yet, just like you were saying, although there is this mounting evidence pointing towards neutralizing antibodies as the potential correlate, we still don't know exactly what part in protection is playing the cellular mediated immunity, whether we can use binding antibodies, so that would be the total IgG produced in lieu of neutralizing antibody, which are more difficult to measure, more cumbersome to measure, whether they are correlating well enough that we can use binding antibodies. And also, we also don't know whether the same correlate would be applicable to all platform, would be the same for a messenger RNA and for an adenoviral vector vaccine, or do we need to establish different correlate per platform, more likely, or per age group? We don't know all these answers, and we will need to wait for more data to be generated to get there.
Thank you.
Now, question number 8. Can the process for flu vaccine strain change be applied to COVID-19? I think that we had a very large response, and 93% of the people have chosen correctly that this is under discussion. There are differences among the two viruses, how they behave, but the experience with influenza is extremely useful, and most of it, if relevant aspects, can be considered applicable to COVID-19, and this is under discussion as we speak with WHO and regulators worldwide and other stakeholders, and it's a very interesting and important discussion, and we will need to look what is happening there. And then we have another one. Another question, number 9. Are efficacy data required for approving a variant vaccine? And you can start choosing. Again, even in this case, it's not that we are looking for a right and wrong answer. This is for you to understand what is the position of the EU regulators at this stage, because also in discussing with the international partners, there are slightly different views, although it's important to discuss and then to reach a common position is always very important. But there are so many different aspects to consider. For example, we discussed so far only the variant vaccines for which a counterpart is already approved, the parent vaccine is already approved. But for a completely new vaccine, that is something that we are still discussing. What would that be required? Can we immunobridge to a similar platform if that is already authorized? Or what? Would we require a large-scale efficacy trial? And would that be feasible, considering that several vaccines are already authorized? And that is a discussion that, regardless of the variant, is also occurring at this stage, which is whenever we are not able anymore to have an efficacy trial placebo-controlled because we have so many vaccines authorized that we would not be able to enroll or would not be ethical to administer placebo to vaccine recipients. Can we have another design, or can we skip the requirement for efficacy trial and go directly into immunobridging? So that is the sort of questions that are still under discussion. And I see now that 40 people have voted for this question. and 68% are correct. It depends if it's a new vaccine or a modification of an authorized vaccine, because if it's a modification of an authorized vaccine, immunogenicity data are enough. But as I just mentioned, for a new vaccine, it is still under discussion. We need to keep in mind what is feasible and what is possible also from an epidemiological perspective. If the vaccines are already authorized, or if the variant has already taken over completely the parent strain. So there are a lot of considerations in this. In this case. So we haven't decided yet. It could be that the 25% are correct in some parts, and definitely we wish all very much that the correlative of protection is identified soon, but at this stage we are not there yet. I think I have no more questions in Slido, and maybe we can go back to the presentation. Yes. And here is my summary slide. So what I try to tell you today is that existing processes to support vaccine development and data evaluation have been accelerated by EMA and adapted to the specificity of the pandemic scenario to support faster approval and availability of vaccines across the European Union. And that global collaboration with regulators, especially under the ICMRA umbrella, which is the hub of international collaboration for global regulatory authorities, and at the level of the scientific community, has proven crucial for a rapid and effective contribution to public health. EMA has established stringent criteria for phase 1, 2, and phase 3 trial design and stringent requirement for vaccines approval, which is very important to make sure that despite the acceleration and the streamlining of the procedure, the standards for quality, efficacy, and safety have not changed and have not been compromised. We have dedicated resources to especially working on COVID. We have flexible procedures. The rolling review and the conditional marketing authorization are two key examples for evaluation of priority vaccines for COVID to avoid delays and bottlenecks. And we also have started to discuss requirements and procedures for variant vaccines. COVID-19 vaccines are only approved when the demonstration of efficacy and safety is based on compelling data before they are used in the general population. And an important, very important part of the discussion is focusing on post-authorization commitments. Manufacturing and trial complete data sets are imposed as obligations to manufacturing authorization holder to be submitted post-approval within specified timelines. But in addition to that, there is a robust system for safety and effectiveness monitoring of vaccine use in real life in the EU that was already present before COVID, but is now being strengthened with additional funding and additional resources and discussions as we speak for an overall robust network for this type of data to support approval and post-authorization. And that concludes my presentation. So I'm happy to take any final questions, clarification, and if you have anything else, I'm happy to respond in writing. Here you have my email. Thank you.
Thank you very much, Manuela. This is fantastic and a very good presentation. I think a lot of people were very interested. We don't have any more questions in the chat, but let's give, we still have 10 minutes. And sorry for rushing you earlier. I thought we were finishing half an hour ago, so that's good because we have more time for discussion. And feel free to ask new questions or to take the floor if you want. Any more questions? Any more comments? Thank you very much for the participation in Slido in any case, because I think it shows the interest and the understanding of what has been presented, and that's what we're trying to reach and obtain. So. Last chance to ask question to Manuela, who knows everything about vaccines.
Oh, okay. I have one question. One question after the last week conclusion from the Pharmacovigilance Risk Assessment Committee on the connection of the thromboembolic events with the AstraZeneca COVID vaccine. So we are aware of the fact that the summary of the product characteristics is updated and will be updated in that sense. But our question is, it is now up to the member states to potentially decide on the use of the vaccines considering the age groups, and is that within the domain of the national medicines agencies or perhaps NITED, or do you know how will the member states, if any, decide on the potential changes of the use considering the different age groups for this vaccine?
Yeah, thank you very much. At this stage, although the review is ongoing, so we need to see what happens next and how the situation will change in the future based on how the available evidence will be coming and will be assessed. However, at this stage, as you said, there has been an update in terms of Section 4.8. So the new adverse event is being listed, but there is no change in terms of overall benefit risk balance for this vaccine. But you are right in saying that member states remain free to update their vaccination recommendation. That is to decide which vaccine should be used in a specific age strata or in a specific risk group. That is their prerogative and is a discussion that is ongoing. I think already some of the member states have decided to use AstraZeneca in the age group above 60 years of age, if I remember correctly. But this is indeed their prerogative based on the data that they have available and based on their overall strategy, because member states can also look into cost effectiveness aspects, comparative data that they might have. So the situation, they can change the vaccination recommendation. That is their remit, is not in the EMA remit. We do not enter into that discussion, although we discuss with the national competent authorities, with the public health authorities that are responsible for these decisions. We discuss with them, we have weekly meetings with them, but we are not responsible to define that. We are only looking at the benefit risk assessment of the product across the age groups.
We have a question about the number of people working on the topic. I think it's a very difficult question to answer, because I can tell you that at EMA we have a large number of people working on vaccines, but not just vaccine, because others are also working more on different topics because they are freeing resources elsewhere. So in practice, I would say we probably have 50 people working on vaccines, I would say, at EMA at any time. That's my guess. But Manuela, you have your own estimate, I guess. In each member state contributing as assessors and rapporteurs and experts, we probably have between 5 and 15, depending on the size of the team. And we also have maybe things that you don't know, that we also have international experts, because we have opened our evaluation for COVID to a number of international regulatory authorities who can join our assessment. And we have open to Health Canada, Japan, Swiss Medic, Australia, WHO, and we have offered it to FDA, but they haven't picked up the offer. But in practice, they have also observed, even if they didn't participate, they have also observed our procedures, especially the one on safety. So you can see that it's a very large number of people working on this to try to make it happen fast for everybody, for the public, because that was an emergency, a public health emergency. So it's really a significant number. And that's so why, although we can probably speed up a little bit our procedures on normal times, we cannot sustain the same rhythm, the same pace as we have had in the recent weeks. I think we will all fall exhausted to this. Manuela, maybe your idea on the number of people working?
I think you probably have a very good estimate. I couldn't say anything different than that. Yeah. but I know that people are dedicating resources and are working day and night. Yeah. So we have a good, because of how the EU system is set up, we have several member states which are swapping products, so that not the same people are looking at the different vaccines, but at least we have a turnover, so that people can rest perhaps a little bit while another team is taking over a new vaccine coming up.
We had a lot of work sharing with member states joining forces because they were not necessarily able to provide the full team of experts. And I think for countries which are candidates and accessing countries, it's quite important to see that we expect people who join our committees and our technical groups to be able to contribute actively, maybe not on all parts of the dossier, but at least on some part of the dossier. And we cannot afford to have people who are passive and just listen and take back home the assessment. We need active members. And that's also what we were trying to achieve through this training, is to make you active participant. That's also why we encourage you to respond to Slido, to make comments, to put your comments in the chat. We want active people. Don't feel afraid. You may not be expert on all areas, but you have your own expertise, and this is always welcome. So this is really an important message from our side.
Yes, that is correct. Yeah, and we have had products which were assessed by a multinational team. So member states can have strength in quality aspects, and they can couple with another member state that is taking care of the clinical aspects of the dossier, for example. So that has helped a lot, this option, to ensure that everybody's contributing and not the same player. Yeah.
Thank you very much, Manuela. I think we are reaching the end of our session. It was a fantastic one. Thank you very much. I think you've been really well spoiled today, all of you, to be able to attend such interesting sessions, very different. This morning we had how to assess a new product, then we had how to assess a generic or biosimilar, and now we've seen vaccines. There are lots of common elements about quality, safety, and efficacy, about the use of the conditional marketing authorization. And so you've seen across these three presentations a number of elements that are common, but everything relies on the network working together and contributing to the assessment. So it's quite important to understand that we are all colleagues, we work together and for the good of the patient in Europe and beyond. And our assessment, for example, of the AstraZeneca vaccine has been used by WHO as part of this open procedure where we have invited them to join. And they used that assessment to assess more quickly and deliver the emergency use listing for the AstraZeneca vaccine. So the CHMP, ETF CHMP work, the rolling review, all the process that has been set in Europe has been actually delivering to everybody else in the world. So I think we can be proud of the work that's been done. It's been hard, but it's a good. Thank you again very much, Manuela. This was very interesting. Thank you, everybody, for your participation. Please remember that we will need a list of participants. So all those of you who are in a group, in a room, we would recommend that you collect the name of the present so we can have a full list. Don't hesitate also to send us your email at emainternational@ema.europa.eu so we can send you the slide. And when the recordings are available on the EMA YouTube channel, we will also inform the participants. Thank you again, Manuela. Thank you, Clément and Sandra. And I think we can close our session, our webinar. Thank you very much. Bye-bye.